volume is low. Hold on, let me turn this up. All righty. Good morning, everyone. Today is Wednesday, October 21st. Looks like some of you, at least, recovered from the exam and have worked your way here. We are on to the fourth section of the class. I want to say that I have started grading the lab exams, and to be honest, so far, it's not pretty. It's a pretty mixed bag of material, but it is, some people were really challenged with this, and that concerns me, because what I will tell you about this next section is it has a similar amount of anatomy, but it has a lot more physiology. So I'm hoping the lecture exams were better. And again, I'm only about a third of the way through the lecture, through the lab exam, so I'm hoping they will improve as well. But again, this format is a little challenging, so you really have to work, and you really have to take advantage of the resources that are available to you. And we'll talk more about some of those things that can help you to be successful in this at the end of lecture today. We are on to our fourth section, which is the muscular system. Towards that end, today we'll be talking about our muscle tissue and talk some muscle histology. And then starting on Monday, we're going to be going all physiology in lecture and all anatomy in lab, kind of similar to what we did with the skeletal system. You have 2, 4, 5 assignments due in this section. You have a labster muscle tissue, which is due on Monday. Again, remember, for those labsters, you have to complete the entire activity, and you must get at least 80% correct for full credit. You have 2 unit reviews, your unit 10 and your unit 11. You have a Physio X. You remember how much fun the first Physio X was, and this one has 7 activities. So that means it's even almost twice as much fun as the first one was for everybody to be able to do. And the other thing that you have is another activity. It's not quite a review the same way that the skeletal system was, but it is the same type of concept where it's kind of taking the information that we're using and putting it together in the body, and that is a muscular activity handout. This will be graded for correctness, and we'll talk more about that on Monday. When we were talking about the origins and interest. Actually, we'll talk more about it today at the end of class, when we talk more about what you need to do to be successful on that. The one bright side spot I see on the horizon for this section is that Wednesday, November 11th, is a holiday. Again, the way that the schedule worked out, that would normally be our testing day, because we will have covered all the material at that point. But because of the holiday, our exam gets moved till Monday. What that means is that Monday the 9th is the last day that you are receiving new information, and that gives you a full week to study and master that, and you are going to need it for this section. So it's one of those things that on the one hand worries me, because I see people thinking that well, but see, here's the problem, Allison, with that is that I fear too many people say, oh, I have a whole week to study this, so I don't have to be working on it now, because once he presents it all, then I have a week after that. That week by itself is not going to be enough. You need to keep up on the information. You need to be consistent with the information. You need to really be diligent, and because there's a lot of it. And I think that this material is a little less intuitive in the anatomy standpoint, because there's so many layers to the material that you're responsible for. Last time it was just, what is this bone? What is this bone feature? And that was challenging for many people. This one's going to be even more challenging. And so again, so the one good side is you do get that extra time, but make sure you are using all of your time wisely. And we'll talk about a lot of the tools that you may not be necessarily using yet, but that can definitely help you to be successful moving forward for that. All right, and so then, like I said, the 16th will be your lab and lecture exam. You've already taken 3 of these. You know what to expect from those. So that's the game plan. Any questions on that? One silly thing I do want to point out, just again, notice that Wednesday, the 28th is not on the schedule. That does not mean that we have that day off. That does not mean that it is a holiday. All that means is you don't have an assignment due on that day. Although it's usually a good guess that if there's a day that you don't have an assignment due, that's usually a good day for a quiz. So you might want to keep that in mind as we work towards the rest of this schedule as well. All right. Questions on any of that? All right, let's dive into the material then. Starting first with our discussion of muscle tissue. So, just like we've talked about for all of our other tissues, we talked about epithelial tissues, connective tissues, all muscle tissues. And really, remember, this is kind of key to what we're talking about here. When I say muscle tissue and I say all muscle tissue, someone remind me again how many different flavors muscle tissue comes in?
3.
3. What were they again?
Smooth, cardiac, and muscle, skeletal.
Excellent. Smooth, cardiac, and skeletal. Excellent. So when I talk about functions of muscles, I'm really talking about all muscle tissue, smooth, cardiac, and skeletal. What are some of the functions? Tell me what muscle tissues do for us.
Generate heat.
Excellent. That is spectacular. Generate heat.
Allow you to flex and extend.
Yeah, movement. Okay, I like that.
Locomotion.
Right, absolutely, right. We can kind of put all of that into the category of moving our body through space.
Maintain posture.
Excellent. Helps us to maintain our posture. You know, that's something that we totally take for granted, right? If I were standing here like I normally would be in the classroom, right, one of the things that I would be able to do is I would be able to, you know, hold my hands out, close my eyes, hold my breath, right? And I wouldn't move, right? And the reason I wouldn't move is because I have all these tiny little minute contractions that are taking place in the muscles of my body to maintain that balance and maintain that posture. And we totally take for granted how complicated that process is until it is impaired. Like after that third shot of Jägermeister, then suddenly it becomes a little bit more challenging to be able to stand there without wobbling back and forth, right? I want to go back also to this idea of generating heat as well. Our muscles are able to generate heat because inside of our muscles, we have something called a motor. I think we talked about this once before, but this is a good thing to do again. What do I mean by motor?
Able to move, muscles that provide movement.
Moving
Well, you're right.
moving substances within the body.
Well, true, that is another function as well. But again, that isn't necessarily what a motor is. So you're absolutely right. When we think of movement, we think of movement of our body through space. But also, if I stand here and hold my breath, there's plenty of movement going on. For instance, there's the contraction of my stomach to move that cheeseburger that I had through breakfast, pumping of my heart to move the blood through me. So absolutely, one of the other types of movement that our muscles allow is that propulsion through hollow organs. Right, so we have things like the pumping of our heart, or we also have things like this rhythmic contraction that takes place within the smooth muscles of organs like our stomach or our small intestines. And that rhythmic contraction of the smooth muscle has a special name. Anyone know what the special name of that rhythmic smooth muscle contraction?
Peristalsis.
Peristalsis, excellent. Peristalsis, spectacular. Yep, absolutely. Those are definitely functions of the muscle tissue. But again, I want to come back to this concept of a motor. What is a motor?
Motor is like an engine.
Okay, and what does an engine do?
Engine produce heat.
Well, is that really the function of your engine, to produce heat?
It means makes you move.
Yeah, exactly. So again, normally, and I have to work on some analogies because normally when I'm sitting here in the classroom, I point out that many of you got here today because of a motor, right? Because the engine in your car. What a motor does, I thought we had talked about this already, but apparently not. So this is good. I'm glad we're bringing it up here. What a motor does is it converts chemical energy into mechanical energy. All right, there you go. Caloric energy into kinetic energy. Daniel's even fancier than saying it than the way I am. Absolutely. That is absolutely correct. However, like the engine in your car,
Can you say, sir, like potential energy to mechanical energy?
chemical energy is a form of potential energy, but you got to be careful, right? If I'm holding my iPhone up here in the air, it has a potential energy in it as well, which is why when I release it, it drops. But that wouldn't be chemical energy. So you're right, chemical energy could be considered a form of potential energy, but I wouldn't use the term potential energy interchangeably with it. Okay? It converts chemical energy into mechanical energy. Your car does that by converting gasoline into propulsion. Well, your muscles, what's the energy that your chemical energy that your muscles use to convert into mechanical energy?
ATP?
ATP, absolutely, ATP. Allison, you're right. Nutrition has to be converted into ATP, has to be broken down to make the ATP. But really what the muscle uses is the muscle uses that ATP directly to break it down to produce mechanical energy. And like your car, it isn't 100% efficient. I think I saw someone mentioned it earlier about how the heat, yeah, Allison mentioned how the heat is a byproduct. Your engine in your car is not 100% efficient, which is why when it gets you to the grocery store, you don't thank your car by getting out and hugging it and putting your head on the hood, right? Because of all that heat that has come off. It's the same thing with our muscles. In fact, some of the muscles of our body are so inefficient that as much as 75% of the energy is lost as heat. Now, would you necessarily want that for the engine of your car? Would you want 75% of the gasoline that you were putting into it to just be lost in the form of heat instead of moving your car forward in space? No, you definitely wouldn't want that. But as someone mentioned, it helps us in that it helps us to generate heat, to maintain our body temperature, right? So when we get cold, we shiver. And the reason we shiver is the contractions of those muscles give off heat as a waste product of that process. And that helps us to maintain our body temperature. Excellent. What else? What else does muscle tissue do for us? I can think of at least two more.
Stabilize the
Or
joint.
Yeah, absolutely. Not only does it help to maintain our posture, but it helps us to stabilize our joints. I know I tend to talk with my hands a lot when I talk, but right, if I was talking and not using my right arm, my humerus doesn't just slide out of the scapula because I'm not using it. I have muscle tone in there that is going to help to stabilize the joints. So both this maintaining posture, and actually let's do it this way, and stabilizing the joints, we really take advantage not so much of the muscle physically moving us through space, but we take advantage of muscle tone to actually maintain that tension, maintain that posture, maintain that stability, and provide protection. Right, that muscle tone does that as well, right? Up here we have bones, here we have bones, but what about our stomach? What about our small and large intestine? Are those protected by bones?
no.
No, we have all these layers of muscles oriented at different orientations, providing what we actually call the abdominal girdle. And that abdominal girdle provides that protection for many of the organs in our abdominal pelvic cavity. All right. All of these are important functions of muscle tissue, but there's at least one more that I can think of off the top of my head. And that's, all right, summer's right around the corner. All right, I'm
Support
sorry.
soft tissues. Support soft tissues and joints.
Sure. So yeah, stabilize the joints, provide protection, support soft tissues. I like that. Absolutely. So summer's right around the corner. Okay, no, it's not, but let's pretend that it was, right? So we all have to get into our bikini bodies. So we do something like the popcorn diet, right? The popcorn diet is where you get one piece of popcorn for breakfast, one piece of popcorn for lunch, and then a reasonable 3 pieces of popcorn for dinner that evening. Right? If this was your diet for a couple weeks, would you lose weight as a result of that? Yes, of course, absolutely. It would be sad, but you would lose weight as a result of that. However, would it be the fat that was just melting off of you?
Water weight and muscle.
There you go, exactly. The other thing, right? When our body is starved, fat is actually a valuable resource that your body holds on to. Muscle, on the other hand, is an incredibly dynamic tissue that is chock filled with proteins. And so, as Haley pointed out, what happens is when we're deprived of those nutrients, our body is much more likely to break down those muscle cells to get the resources that it needs out of it than it is to do it out of the fat. So it is actually a nutrient reserve. Right? Muscle tissue is very, very dynamic. Right? It's truly that use it or lose it type of mentality. And if we are starving ourselves, then yes, you're going to end up losing water weight, but you're also going to break down your muscle as well. So it acts as a nutrient reserve. I feel like there's another one, but I can't think of it off the top of my head. So let's cheat and look at our list. Reducing body movements, stabilizing body positions, moving substances within the body. Oh, that's the one thing I wanted to say. The way it moves the substances
I've
through
already
the body.
mentioned this, sir.
I'm sorry?
I've already mentioned this.
Yes, I am aware of that. But what I wanted to say was that the way it does that is by changing the volume of the organ, right? This works with the hollow organs of the body. And when we change the size of that hollow organ, we change the volume. And when we change the volume, what do we change? If you change the volume of something, what happens in response to that? You change what makes the world go round?
Pressure.
Pressure. You change the volume and you change the pressure. And the same way that things move down a concentration gradient, things move down a pressure gradient. So if in an organ, one side squeezes, the volume gets shorter, the pressure goes up, and the food is moved to the next region because it doesn't want to be in that higher pressure region, so it moves to a lower pressure area. So it is those pressure changes. Again, pressure is what makes the world go round. Great question. So the reason we put it in the muscle is when you get an intramuscular injection is really twofold. The reason for it and which muscle you get it in is influenced by basically two factors. First, you need a tissue that is large enough to take the bolus of medicine, and muscle has a nice density to it. But you also want it to have a good enough blood supply to it where it's able to distribute that medicine at a reasonable rate throughout the body. Because again, if you wanted it all in the body at once, you would put it into a vein. In this case, you want a blood flow to it that is vigorous enough that it is going to be able to distribute it, not necessarily all at once, but you don't want it just sitting there either. So those are really the reasons why we give that. True, some medicines need to be put in fat or subcutaneously. Again, there are things like that. Not always bigger muscles, because there's the other thing that you have to worry about with the muscles are also heavily innervated. So really the three things that they look for in a muscle when they're going to give an intramuscular injection is it has to be large enough to accept the bolus of medicine. It has to have an appropriate blood supply to be able to distribute the medicine. And there can't be any large major nerves in that area that you could accidentally hit with the needle. So where are the two muscles that are most commonly used for intramuscular injections?
Gluteal maximus.
So the deltoid, absolutely the deltoid. And I will say the gluteus maximus is great in the movies. Fun, it always gets a good laugh in the movies. But in reality, it isn't actually the gluteus maximus. The reason they don't use the gluteus maximus is that the sciatic nerve passes through that area, and that is an area of risk. So where they actually go is into the gluteus medius. Yeah, exactly. You find the hip, you go down a couple inches from the hip, and it goes that way. Now, I saw someone also said the thigh, and the thigh is true in children, is one of the primary locations you do it in children because their muscles are smaller, so you need a larger muscle mass. But in adults, it's typically in the deltoid or the gluteus medius. All right. Excellent. Support soft tissue and joints. body temperature, nutrient reserve. Okay, excellent. So we did hit them all. Spectacular. So those are the functions of all of our muscles. And one of the cool things about muscles is really they're pretty much a one-trick pony. They have one active process that they do. What is the one active thing muscles do?
Constrict.
Contract, absolutely. So they're able to do all these things, and being one-trick ponies, they are able to contract. Now, let's talk about the properties. Again, we are still talking about all muscle tissues at this point. Just like we talked about properties or characteristics of epithelial tissues, they're superficial. Properties or characteristics of connective tissues, they have matrix. We need to talk about the properties and characteristics of all muscle tissues. The first is that they're irritable. What does it mean to be irritable?
Receive in response to stimuli.
Exactly, absolutely. You receive a stimulus and you respond to that stimulus, right? You are a 49ers fan and Garoppolo, as beautiful as he is, throws yet another interception and you cry as a result of that, right? You receive a stimulus and you respond to it. That's what it means to be irritable. Although my favorite example was my grandmother. My grandmother was an incredibly loving individual, right? She would give you anything. She was the sweetest, nicest, kindest person in the world, unless her stories were on TV. If her stories were on TV, you didn't mess with grandma. If you talked during grandma's stories, she would throw her shoe at you, right? So again, she would receive a stimulus, hear you talking, and whoosh, she would hit you with that shoe. So that's what it means to be irritable, to receive a stimulus and respond to it. In the case of muscle tissue responds to a stimulus by producing an electrical signal. That electrical signal is a big, huge positive signal used for communication. And does anybody know what that would be? What the name of that big positive electrical signal would be?
Membrane potential?
Action potential.
Action potential. You're close, but yeah, absolutely. Action potential, absolutely. An action potential. So we can also say not only is muscle tissue irritable, but it is also electrically excitable. When it is stimulated, it responds to that stimulus, and it does it by producing a big electrical signal called an action potential. This is similar to what a nerve does in our nervous tissue. So we can be more specific by saying it produces a muscle action potential. So it is going to produce a muscle action potential in response to a stimulus. Now, when that muscle action potential is produced, it stimulates the muscle to contract. Right? What does it mean to contract?
Make shorter, the muscles get shorter.
Right, it changes the length of the muscle, and that change in length generates a force. Oops. Right, it generates a tension. You can say it either of those two ways. It's going to generate a force. It is going to generate a tension. And this is an active process. What does that mean?
Requires energy.
And when we're talking about cells, that energy is in the form of
ATP.
Yeah, absolutely. This uses ATP. This is an active process that uses ATP. Right? It is able to change its length, and when it changes its length, it generates that force, it generates that tension that we can use to move our body through space. And again, this is an active process. And make a point of emphasizing this because our third characteristic of muscle tissue, oh, I don't have one, do I? Shucks. Our third example of a property is extensibility. What does extensibility mean?
It can stretch.
It's able to stretch and extend.
Okay, well, be careful. It's not that it is able to stretch itself, but it is able to be stretched. One of the key things about this is that when we have this extensibility, it is able to be stretched without damage. Right?
And then relax.
Well, not necessarily so much relax, because again, relax, I think of activity, right? I don't have a rubber band, but if I had a rubber band, one of the things that I would be able to do is take this rubber band and I would be able to stretch it out, right?
Elasticity.
It would not be damaged by that, right? However, are there limits to how long I could stretch it? Could I stretch it far enough to damage it, to break it?
Yes.
Yeah, of course, absolutely, absolutely. So there is some limit to that extensibility. But here's the other thing. If I took that rubber band and I set it here on the countertop, is it ever going to stretch itself out on its own?
No.
It's not going to be able to stretch itself out on its own. So this, again, is a passive property. It can be stretched. So it is stretched by something else. It can be stretched without damage. Right, and that's what it means to be extensible, able to be stretched without damage, but it is a passive process. It's not stretching itself out. That's an important thing. When we talk about this muscle right here, anybody know what this superficial muscle here on my upper arm would be?
The brachius.
Biceps.
Biceps brachia, there you go, absolutely. When I contract my bicep brachia using the active ability of its contractility, it gets shorter, and as it gets shorter, it pulls my elbow and my forearm so that my arm flexes. If I wanted to extend my elbow, does my bicep brachia push my arm away? Is that how I'm able to extend my elbow?
No.
No. Instead, what happens is I contract my tricep brachia. And as I contract my tricep brachia, notice my bicep brachia is stretched out. It's not damaged by that, but it's not using ATP. It is just being stretched, right? However, if I hyperextended my elbow, could I overstretch my bicep brachia and damage it? Sure, probably.
Yeah.
Yes.
Yeah, but it can be stretched without damage. That's the key to that, all right? But as you guys also have pointed out, when I stretch out that rubber band and let go, what happens to it?
Recoils.
Yeah, it goes back to its original shape. It recoils back to its original shape. And that characteristic is what we call elasticity. If I stretch out the rubber band and let it go, it goes back to its original shape. If I take the rubber band and I scrunch it up into a ball and then open my hand, it goes back to the original shape. That's that elasticity. When you release the tension from the muscle tissue goes back to its normal length and its normal shape. And this is true. All the examples we used, we talked about skeletal muscle, but remember, this is true for smooth muscle, this is true for cardiac muscle, this is true for skeletal muscle. All of our muscle tissues have these four properties: irritability, contractility, extensibility, and elasticity. And again, let's be specific. Elasticity, passive or active process?
Passive.
Passive. Absolutely. Again, it doesn't use ATP. So the only active ability of our muscle tissue, the only time it's actively using ATP is when it's contracting to change its shape, shortening to generate that force, to generate that tension. All right. Questions on that?
So as you mentioned, sir, this is the for the muscles. We have also when the contracts, they receive the signals or the stimuli. So we call it muscle action potential. So this is the only difference with the nerve action potential, or the action potential is produced in the nervous system, or the in the axon, or
So They are very, very similar. And one of the reasons I like doing the muscle tissue first is the muscle action potential is kind of a baby action potential. It's an opportunity for us to get some familiarity with the action potential so that when we get to the nervous tissue next, we get to learn a little bit more about it and how more complicated it is. They are fairly similar in their function, but not identical. But obviously we have to talk about and learn about both before we can compare them. And we will get that when we get to the nervous system. Okay.
Good.
Excellent. All right. So these are characteristics that all muscle tissues have, just like we talked about epithelial tissues and all the characteristics of them, and connective tissues and all the characteristics of them. But we also know that there are specific types, and those specific types have their own unique characteristics. And so that's what we need to do next. What we need to do next is we need to talk about the three types of muscle tissue and talk about their characteristics. All right, and let's do this way. So remind me again of the three types.
Skeletal muscle.
Skeletal, cardiac, and smooth.
All right, skeletal, smooth, cardiac. Thank you. Excellent. All right. Let's talk about skeletal muscle first. Why is it called skeletal muscle? Where is it located?
Top of your
Within
body.
the muscles.
Okay, well, they do make, well, all of these make muscles, so we want to be careful. So where would you say, okay, I like that. Allison's got it. Most of our skeletal muscles are attached to bones. Of course, what's the key word in that sentence?
Most.
Most means not all. For instance, there are some examples of some facial muscles where the muscle actually attaches from one muscle to another muscle, or they may actually connect from muscle to skin, for instance, allowing us to move our face and give facial expressions. But most skeletal muscle is attached to bones, which is why it is called skeletal muscle. It is the largest of our muscle cells, and it has a cylinder shape. So these are big, long, tube-like muscle cells. In fact, a single skeletal muscle cell can be as much as 1 foot long. You have some skeletal muscle cells in your body that are about 12 inches in length. So they are the largest of all of them. They also have another unique characteristic we haven't seen too much up to this point in time. They are multinuclear. What does that mean?
Have multiple nuclei.
Exactly, they have multiple nuclei. One of the cool things about our skeletal muscle is that it is chock filled with proteins inside. So many proteins so perfectly aligned that there's no room for the nucleus on the inside. So what happens is the nuclei are kind of pushed out onto the periphery. They're still inside the plasma membrane, don't get me wrong, but the nuclei are all located on the periphery of the cell. And so there are many nuclei located on the periphery of this cell. As I mentioned, they're chock filled with proteins, with perfectly aligned. Myosin and actin are definitely a couple of them, although there are literally dozens of these proteins that are perfectly aligned. And they're so perfectly aligned that it turns out that there are some regions of this muscle cell that have a higher concentration of proteins and others that have a lower concentration of proteins. And because of that, it gives the muscle a striated appearance. And what does it mean to be striated?
Linear or with the lines.
Yeah, it has stripes, absolutely. So there's some areas that have a higher concentration of proteins and some that have a lower concentration of proteins. And so the muscle cell actually has these stripes that run perpendicular to their longitudinal axis. So the cell has these stripes. It is striated in its appearance. All right. Now, skeletal muscle is what we commonly think of when we think of our muscles and the things that we move through space. And as such, it is voluntarily controlled. Right? I can move my fingers of my hand all I want. But if I cut my hand off, leave it on the countertop here, even if I provide it with all the oxygen and the nutrients that it needs, is it going to be able to run off and live a happy life? Right, we can call it thing.
No.
No, of course not. Absolutely not. It is voluntarily controlled, and it is voluntarily controlled by what we call our somatic motor nervous system. Somatic means
Body.
Yep, body, exactly. So it's our somatic motor nervous system that controls and gives us that voluntary control. And I know you know one other thing about this as well, because if I decide I want to wiggle my right finger here, where do I actually make that decision?
In brain.
Yeah, but where in the brain? Over here on the right side?
The
There you go. Alina's got it on the left side. If I want to move my right finger, I make that decision with the left side of my brain. It actually has to cross the midline to get over there. Anybody remember what that's called? Where you have information that crosses the midline? If it was on the same side, it would be ipsilateral, but since it crosses the midline, that means it must be, there we go, contralateral. We have contralateral control of our somatic motor nervous system that controls our skeletal muscle. All right, one more thing, regeneration. We learned about the concept of healing. You have two choices, regeneration or fibrosis. Which one is used to heal skeletal muscle? Regeneration or fibrosis?
Fibrosis.
All right, so I've got one vote for regeneration. I've got one vote for fibrosis. Which one's correct?
Fibrosis.
No, you're actually both correct, right? Our skeletal muscle, if while lifting weights, right, to impress my wife, I damage my arm muscle, right? Is it going to heal as a result of that? Yeah, likely, and it's going to regenerate and it's going to be fine afterwards. However, if a bear with an axe takes a big, huge chunk out of my shoulder, am I going to be able to regrow all of that tissue? No. So skeletal muscle has a very limited regeneration, right? Oops, if I spell it right. Limited regeneration. It is small, minor injuries can be regenerated, but if large portions are damaged, then it's going to be replaced with scar tissue. And is that scar tissue going to look the same or function the same as the muscle tissue that was there in my shoulder before?
No.
No, of course not. So again, so that a limited regeneration is an important characteristic of it. All right, excellent. Let's do the same thing now with smooth muscle. Where is smooth muscle located?
The walls of your hollow organs.
I like that. Within the walls of our hollow organs. All of it?
No.
Most is within the walls of our hollow organs, absolutely, but not all. Can somebody give an example of where we might find smooth muscle that isn't in the wall of the hollow organ? Well, I would argue that your nose is a hollow organ. If only, there we go. If only in class we had already learned about a location where we have smooth muscle that functions to maybe make the hairs of our arms stand up on end. Alina's got it. Our rector pili muscles are examples of smooth muscle that is able to not be found inside the wall of a hollow organ. Excellent. Smooth muscle is our smallest muscle cell. They are very tiny muscle cells, and they are typically arranged in layers. So we have these layers of the cells all interwoven with each other that then are able to work together in that fashion. Smooth muscle has what we call a spindle shape to it. A spindle shape means that there is a large centrally located body, and then it has these two kind of tapered ends to it. Obviously, these are not drawn to scale with each other, but you get the idea vaguely of what a spindle shape looks like. Here, actually, let's cheat. There, pretend that was rounded. That's kind of what a spindle shape is. You have a large centrally located body and then a tapering tails on the two sides. Right? And I need to move this down. Smooth muscle also has just one centrally located nucleus. So just one big centrally located nucleus in the center of it here. And why does it get the name smooth?
Because no straights.
There you go. No striations. Right? It uses, it has a different arrangement to its proteins, and it has no stripes. This is going to be important because what we are going to learn is that precise arrangement of the proteins in the skeletal muscle is what forms the functional and the structural units. It is going to be what allows the muscle cell to contract. And if smooth muscle doesn't have those striations, then that means smooth muscle must contract using a different mechanism than our skeletal muscle does. Excellent. And do we voluntarily control our stomach?
No.
Or our rector pili muscles?
It's involuntary.
Exactly. So we get that involuntary control. So involuntary controlled, to be precise. And so for that, it is involuntarily controlled. And again, if I rip my stomach out and I put it on the counter here and I provide it with all the oxygen and the nutrients that it needs, is it going to happily be able to contract away at that cheeseburger you had for breakfast?
No.
No, absolutely not. But we don't voluntarily control it. So it is controlled instead by a branch of our nervous system called the autonomic nervous system, and also by hormones. There you go. Excellent. And what about smooth muscle's ability to regenerate? Do you think smooth muscle can regenerate?
Yes.
Yeah, in fact, it actually has the highest rate of regeneration. All right, whoever's got their TV on really loud has got to be careful about that when they turn their microphone on, please. So it has the highest rate of regeneration of all of our muscle tissue types. All right, questions on smooth muscle? All right, then let's talk about cardiac muscle. Where is cardiac muscle located?
Cardiac in the heart.
Only the heart.
Excellent, and that's what I wanted to hear. Heart and heart only. All right, so no most here. It is located one place and one place only, in the heart. Notice it's neither the largest or the smallest. It's kind of in between smooth and skeletal muscle. And the same is true for its other characteristics as well. It's kind of this interesting mishmash of both of them. Obviously, it is intermediate in size. But cardiac muscle has what we call a branching shape. So what happens is we have this cell, right? That, here, let's just make it red like the rest of them. Yeah, it can have kind of a somewhat cylinder shape, but what happens is the ends of it can be branched. So it can communicate to multiple cells, and that branching can occur on one or both ends. So it has this branching shape. Like smooth muscle, it is uninucleated with just one centrally located nucleus. But like skeletal muscle, it is striated. So it has that same perfect arrangement of proteins forming those same structural and functional units. So the same way that skeletal muscle contracts is going to be similar to the way that our cardiac muscle contracts. So it is striated, does have that perfectly aligned proteins the same way. When it comes to control, is it involuntary or voluntary?
Involuntary.
All right. And like smooth muscle, like cardiac muscle, like skeletal muscle, if I rip my heart out, put it in a petri dish and provided it with all the oxygen and the nutrients that it need, would it contract away?
No.
Yeah, turns out it
Yeah.
actually would. Absolutely. Our cardiac muscle has something special about it. It is what we call autorhythmic. What this means is it is actually able to produce its own action potentials. And of course, when it produces its own action potentials, it is therefore able to contract. So indeed, if you put a live heart in a petri dish and provided it with all the oxygen and the nutrients it needs, it would happily contract away. In fact, it would contract away at a rate of about 100 beats per minute. Is that your resting heart rate right now?
No.
No, unless you hit Dutch Brothers on the way to class today and got that quad shot, hopefully your heart isn't beating 100 beats per minute. So it is actually able to produce its own action potentials. However, we are able to modify its behavior. So we don't control it, but what we do is we are able to modify the activity of the heart. And we do that in two ways. Do we do it voluntarily?
No.
No, yeah, you don't have to say, all right, I've got these 3 flights of stairs I have to run up, heart beat stronger, faster, so that I can go up those stairs. No, it's involuntarily modified. So we, again, modify it involuntarily.
It responds to the stressors we give it.
True. So you're right. We can take advantage of that. We can trick it. If we were really to sit here and close our eyes and really imagine what it would be like for a bear with an axe to walk into the room, look you in the eye and say, I'm going to kill you. And you really sat here for 5 minutes and really thought what that would be looking like, the snot coming out of his nose, right? How is he holding that axe without opposable thumbs, right? You really spent all this time talking about it. Would your heart start beating a little faster in fear for that? Absolutely. If instead we all sat here in the lotus position and went or a cheeseburger or whatever your mantra is, right, then would we possibly be able to lower our heart rate? Yes, but we're not voluntarily doing it. You've got the right idea. We're basically taking advantage of our autonomic nervous system, our fight and flight and our rest and digest processes to influence that. So we're not directly saying, hey, heart, beat slower, right? If we try to put ourselves in a calm meditative state, that increase in our parasympathetic activity would lower our heart rate, but it's not a direct control. And if our autonomic nervous system can speed up the heart, so can that adrenaline, so and hormones as well. So we are going to modify the behavior of the heart with our autonomic nervous system and our hormones. And what about our cardiac muscle's ability to regenerate? Is cardiac muscle able to regenerate?
Yes.
No. All right. It's one of the problems with if you have heart problems, if you have damage to the heart, that heart's not able to heal itself. All right. It's one of the reasons why we have to be very careful about taking care of our heart, because it's not able to repair itself. It will heal via fibrosis. And if we get a big chunk of scar tissue in the heart, can that affect the heart and how it functions and how it beats properly?
Yes.
Absolutely. And then you have to either get a pacemaker or you get to die. Right, those are your two choices. So absolutely. So there you go. These are the characteristics, and we can go over this again real quickly by looking at the pretty pictures from your textbook. So again, skeletal muscle, mostly connected to the bones, but not all. Striated, voluntary, multinucleated, controlled by the somatic motor nervous system. Our smooth muscle, mostly in the walls of our hollow organs, non-striated, involuntary, controlled by hormones and our autonomic nervous system. And then our cardiac muscle, only found on the heart, striated, involuntary, uninucleated, autorhythmic, able to produce its own action potentials, but we can modify that by our hormones and our autonomic nervous system. Like I said, while you're sitting here in class, nobody's heart's probably beating 100 beats per minute. So our rest and digest parasympathetic nervous system is actually slowing our heart down while we're sitting here, right? During the 15-minute breaks where you have to run down 3 flights of stairs to get your cup of coffee and then come running back up, right? Then your heart's beating a little faster during that because of that sympathetic input. So again, we have that ability to modify it. We have that ability to influence, but it has this magical ability to be able to produce its own action potential, which if we have time at the end of this section, we will actually talk about how that occurs. All right, and if you don't worry, if we don't get to it in this class, you will get it in 431 when we talk about the cardiovascular system. All right, and then let's have some pretty pictures to go along with this. What we have here is, where's my highlighter? There. 4 pieces of skeletal muscle cell. So again, this is the boundary of one portion of one of those cylinder-shaped cells. Notice as we talked about, the nuclei are shoved to the periphery of this because inside we have these stripes that are formed by perfectly and precisely arranged proteins. Notice there is a dark stripe and then another dark stripe, and in between that dark stripe is a light stripe. However, if you really look closely, if you have a good resolution to your screen, you will notice that in the center of our light stripe is actually a dark line at the center of that. Hopefully everybody should be able to see that, yes? But if you also look closely, one of the things that you may notice is there is a slightly light colored region at the center of our dark stripe as well. So even though we have a dark stripe and then a light stripe and then a dark stripe and then a light stripe, there are variations within the stripes themselves, right? And that's because these proteins are so perfectly arranged. And look how cool this is. Not only are these so perfectly arranged within a single cell, but notice they are also perfectly arranged with the cells adjacent to them. So all the dark stripes all line up with each other. All the light stripes all line up with each other, giving us this perfect, precise arrangement. And that's where the efficiency comes from. If I took any 4 of you and put you in a boat and gave you 4 oars, would you guys be able to get from one side of a lake to the other? Yeah, absolutely. However, if I put any 4 of you in a boat with oars and put you in a race against a crew team, now again, I'm assuming a real crew team, not Lori Lofton's kids, but a real crew team, then would you guys be able to beat them on the way over there?
No.
No, absolutely not. And the reason for that is that crew team is practiced and works with precise precision. They all are raising their oars at the same time and pulling at the same time and dipping them in the water at the same time. And that precision gives them tremendous efficiency. And so with that tremendous efficiency, we get tremendous strength. And so that's what this precision helps us to allow. This precision allows us to work incredibly efficiently and generate a massive amount of power with these muscle cells. All right. Excellent. So that is our skeletal muscle. Let's take a look, quickly go through a quick review. Yes, go ahead.
So excuse me, sir. One of the things we in the skeletal muscle, we said it's multinucleated, right?
That is correct. So
and we see only one nuclei here, like.
True, technically you only see 3 nuclei for 4 cells, but remember, these are just a small portion of a cell. This cell in this microscope, that image that we see, extends far beyond what we're looking at in here. So there may be another nucleus down here and another nucleus down here. You are not looking at 4 full cells, you're looking at pieces of 4 cells.
Okay,
so that's why you're only seeing a couple nuclei in here, because we have to look really, really close to get this, to get the view that we have here of the stripes. So this is a very high magnification view. So we're only seeing a very small section of that stripe, of that muscle. So that's why we're only seeing a couple nuclei. It's a great question. Excellent. So again, quick review. What is the shape of our skeletal muscle cells?
Cylinder.
Yeah, cylinder, excellent. Where is skeletal muscle found?
Skeleton of the body.
Again?
On the skeleton of the body,
Yeah,
mostly.
attach the skeleton, attach the bones of the body, absolutely. Voluntary, yes or no?
Yes.
Striated, yes or no?
Yes.
Number of nuclei per cell, 1 or many? Many, excellent. Able to regenerate?
Yes.
Yeah, it's got that limited regeneration.
Limited.
Yeah, limited, but a limited, yes, absolutely. It does have a limited regeneration. Excellent. Here, we see a small portion of a layer of smooth muscle. Again, when these spindle-shaped cells are interdigitated with themselves like this, it can be hard to see their spindle shape. So we just see them as this layer. One of the things we'll actually do under the microscope is we will actually look at slides where they've actually used, use needles, the tips of needles, and they tease apart the cells. And as they tease apart the cells, you can actually see the individual cells and the individual spindles of those. So we will get a chance to look at that. But here we see them in an intact sheet. I just gave it away, but what is the shape of our smooth muscle?
Spindle shape.
There you go. Where is our smooth muscle found?
The hollow organs and the wall of the hollow organs.
Mostly in the wall of our hollow organs, absolutely. Voluntary?
Involuntary.
Yeah, so no, not voluntary. Excellent. Striated?
No.
Yes or no? No. Excellent. Number of nuclei, 1 or many?
1.
1. Able to regenerate?
Yes, perfectly, the highest.
Well, not perfectly. Nothing's perfect, right?
Highest, what I'm saying.
Yeah, it is the highest rate of all of them, absolutely. Lastly, we have our cardiac muscle here. Now, there is one thing that I want. Well, let's start with this. What is the shape of cardiac muscle again?
The branched.
Branched, absolutely. So what we see here, for instance, if we look closely, we can see that this single muscle cell is connecting to 1, connecting to 2, connecting to 3 different portions of other muscle cells. So it has this nice branching shape. It has a single body, but then it branches to connect to other cells. And one of the nice things about this branching shape is there is these big spaces within the tissue. These nuclei that you see are not actually the cardiac muscle nuclei. This is nuclei of connective tissue that fills the spaces in between our cardiac muscle. This cardiac muscle needs extra structure, extra support. So it actually has what we call this fibrous skeleton inside of it by having all this connective tissue inside of it, which helps to give the heart its shape and its structure. So this branching shape gives us lots of space for this connective tissue to fill in the gaps there. So we have this great branching shape to it. Excellent. Where's our cardiac muscle found?
Only in the heart?
Only in the heart. Excellent. Voluntary?
No.
Involuntary.
Striated?
Yes.
Yes.
Yes, absolutely. Number of nuclei, one or many?
One.
One. Able to regenerate?
No.
No. Perfect. Excellent. So with that, we have identified the functions of all muscle tissues, the characteristics of all muscle tissues, and specific characteristics of the three muscle tissue types.
One of the questions, sir, we have mentioned it's intermediate in size.
True, you are correct. So yes, again, yes, this one is intermediate in size. Which cells are the largest?
The skeletal muscle.
Skeletal, and then that means the smooth is the smallest. That is correct. Yep. All right, so we have talked about all of them collectively in their characteristics, their functions. We've talked about the three muscle types individually and their specific characteristics. What we are going to do now for most of the rest of the section is we are going to focus on skeletal muscle tissue. So we are going to be talking primarily about skeletal muscle tissue for the majority of this section. At the very end, on the very last day, we'll come back and talk about smooth muscle and cardiac muscle and compare them to what we've learned in skeletal muscle. But for the primary portion, the majority of this class, we are going to be focusing on skeletal muscle tissue. And of course, the organs that are formed. Yes.
Sorry, sir. I thought.
No, you go ahead. What were you going to ask?
No, I talked to my son. Sorry, sir.
Oh, okay, no problem. The organs formed by our skeletal muscle tissue. And what do we call the organs formed by skeletal muscle tissue? Well, what did we call the organs formed by by compact bone connective tissue?
Bones.
Bones. So what do you think we call the organs formed by skeletal muscle tissue?
Muscles.
Muscles. There you go. Exactly. There are skeletal muscles. We had 206 named bones. How many named skeletal muscles do we have?
300 something, 300, around 300 or 600.
There you go, 600, over 600. There are over 600 named skeletal muscles. Do you need to learn all 600 the way you had to learn all 206 bones?
No.
No, of course not. Only about 520, so it's not that bad. All right, I'm just kidding. Hopefully at this point you've looked at the list and you've seen the list that you have is far smaller than 600. So, but the point is our skeletal muscle tissue is what makes our skeletal muscles, and we have over 600. Our goal over the next 3 weeks is going to be to learn everything you ever wanted to know and more about skeletal muscles and how they work, how we're able to do something as simple as flexing our elbow. However, to do that, we have to take that organ that is a skeletal muscle, like the bicep brachia that we talked about, and we have to break it down into its smallest bits. And by smallest bits, I mean we are going to take it all the way down to the proteins. And we're going to look at all the individual proteins and how all those individual proteins interact with each other. And once we know how all those little proteins work together, we can understand how the organelles work together, we can understand how the muscle cell works together, and we can understand how the muscle works together to move our body through space. So we need to take this skeletal muscle, those 600 named skeletal muscles, and break them down into their basic components. Now, each skeletal muscle, again, like the bicep brachia, is made up of somewhere between hundreds and thousands of muscle cells. Now, as we know, skeletal muscle cells are long, thin, cylinder-shaped structures that can be up to a foot long. Because of that, they are often also referred to as muscle fibers. And you will hear me and everybody else use these terms interchangeably. Muscle cell and muscle fiber mean the exact same thing. It's just a big, long, thread-like cell. So it's a fiber-like cell. So muscle fiber, muscle cell, those two words mean the exact same thing. Okay? Now, something like the bicep brachia, is it just a bucket of muscle cells that are all thrown together and poof, you have an organ that is a muscle? No, of course not. These muscle cells need to be precisely arranged. And so they're going to be held together by connective tissue. So we may need many layers of organizing connective tissue to hold these things together. Right? When doing this, we have muscles and muscle groups that are held together by a connective tissue called epimesium would be one example of that, absolutely. And then we'll talk perimysium, endomysium, and the tissue types. We'll talk about all of that organization. But even individual muscles have to be held together. For instance, all of the muscles that flex my elbow have a connective tissue that wrap around them, stabilizing them and holding them in place. All of the muscles that extend my elbow have a connective tissue that wraps around them and holds them in place. That helps to define them, distinguish them, and as they move, it allows the muscles to move back and forth across the surface of each other without damaging them. So we have these fascias that help to distinguish and define the muscles, right? We've actually already talked about one example of one of these fascias, and that was our subcutaneous layer, or the hypodermis. Well, now we get to give a third name to that structure, and that is the superficial fascia. Remember, we talked about how the hypodermis loosely connects the skin to the muscles, allowing them to be able to move freely under the surface of the skin without putting too much pressure and pulling on the skin. But individual muscles, like we talked about, are going to be wrapped around and stabilized by connective tissues as well. And those we call the deep fascia. And like I said, those are the ones that are going to hold, like, for instance, all the muscles of my bicep brachia, or that flex my elbow, and all the ones that extend my elbow and separate them. We can, at some point in this class, if you like, have the deep philosophical discussion of whether or not meat is murder, right? If you want to have that discussion, I'm happy to do that. We can do that at some other time. However, the one thing you cannot deny is that meat is muscle. So this idea of a deep fascia is not something you haven't thought about before. Because, for instance, if you were at home cooking a turkey, Thanksgiving is right around the corner, right? And as you are preparing that raw turkey, the raw turkey leg, are you able to easily remove the meat from that if you were going to try to debone the turkey leg? Would that be something where the muscle would easily come off if you were trying to do that?
Not raw.
Not raw, absolutely not. However, right, back in ancient times, we were able to go to things called Renaissance fairs. And what's the best thing about the Renaissance fair? The turkey legs. You get that big, huge, massive turkey leg. And as you're walking around eating that turkey leg, as you take a bite of that meat, does it all just peel off the bone as one individual piece? No, the individual muscles actually peel apart when that meat has been cooked. And the reason for that is when you've cooked that, it breaks up the connective tissue, separating the individual muscles. So individual muscles can actually pull apart from that when you're eating that. So absolutely. Yes, the beer, the mead, all of those things, the costumes. Again, Renaissance fairs are great things. Hopefully we'll have them again at some point. All right, excellent. So these are the things that hold together functional groups of muscles and allow them to function. But like I said, our goal is to take an individual muscle and break it down into bits and pieces. This is going to be one of those times where there is going to be a massive amount of vocabulary involved in this. So I think at this point, looking at the time, this is probably a good place to go ahead and take our first break so we can come back fresh, because we are going to get hit with a ton of anatomy and a ton of vocabulary, and I want to make sure that we are fresh for that. All right, so any questions before we take our first break? All right, looks like it's 9:10. So let's go ahead and we will restart in 15 minutes. So that will be 9:25. And I will start the recording at that point. All right, any questions on that? All right, I will see you guys in 15 minutes then. All righty. Let's dive back in. So our goal now is going to be to take a muscle, one of those 600 named muscles in the body, and start breaking it down into its bits and pieces. Here, as we can see, for instance, in a cross section, we can see a muscle. That muscle is attached to, of course, a bone. What bone would that be?
Femur.
Femur, excellent. So probably not the bicep brachia that I've been talking about, but maybe the bicep femoris or something along those lines. The point is, we have all of these muscle cells. We see the muscle cell in there, and let's actually grab my highlighter so it's a little easier to see. And they are arranged in this fashion, in this order inside of this. So what we can do is see how this arrangement works. Individual muscle cells. So let's go ahead and draw a couple muscle cells here. 1, 2, and 3. Individual muscle cells. And actually, let's silly to have put them so down so low. Whoops, no. Individual muscle cells, as we mentioned, are fibrous. They're long, thin, length-like structures. And as we talked about, they're irritable. They can produce an electrical signal. And that electrical signal can travel from one end to another, kind of like the wires in your wall. And as you look in the wires in your wall, are they just a bunch of naked copper wires all bundled together and we stick them in the wall and hope for the best?
No.
No, they need to have insulation around them so that each one, when it fires, that signal doesn't spread to another one. And that's what we see here. There is a areolar connective tissue that is going to wrap around and insulate and isolate each individual muscle cell. So each individual muscle cell is going to have its own little special saran wrap of areolar connective tissue. So this is areolar connective tissue. Let's make it purple so that it's consistent. That wraps around each individual one. Now, of course, is this the only place in the body we find areolar connective tissue?
No.
No, we find areolar connective tissue everywhere, which means this areolar connective tissue, we should give a name based on its location. And so again, I will just abbreviate that BOL, but again, remember that means based on location. And that's also how I would ask that on the exam. The based on location, it is called the endomysium. Endo meaning within, mesium obviously referring to muscle. Now, these individual muscle cells with their endomysium need to be held in an arrangement as well. So what happens is, and let's change colors to, we'll use green this time. There is a dense irregular connective tissue that is going to wrap around these muscle cells, stabilizing them in place. This structure is called a fascicle. It is comprised of a dense irregular connective tissue And this dense irregular connective tissue wraps around these muscle cells. So based on its location, we call it a perimysium. Now, these may not be terms that you are aware of, but that you have not heard before, but I know you're aware of this because inside of our muscle, we have many of these fascicles. So I've got 3 muscle cells here, but then there's going to be a bunch more here and a bunch more here, all inside of these. Let's cheat. Move this one up. Move this one up. And move this one up too. Thinking ahead. Not used to that. Excellent. Perfect. Excellent. So we have these fascicles all bundled together inside of the muscle cell. And you may not have heard the term fascicle, you may not have thought of it before, but I know you're aware of it. Because if you're one of those people, those special kind of people who like to take, for instance, like a tri-tip, and you marinate it for 3 days in the bottom of your fridge, then you bring it out on the day you're going to cook it, bring it up to room temperature, get it on that nice hot barbecue to sear the outer surface, then move it over to the side. So you low and slow bring it up to temperature. Then you take it out and you rest it that 20 minutes to bring all the juices back inside. And after all that work and all that effort and all that time, do you just close your eyes and just whack at it at however, just cutting it up into all these random pieces? No, how do you cut up that tri-tip after you've put all that time and love and care into it?
Against the grain.
Against the grain, absolutely, against the grain. Well, that grain of the meat are the fascicles, right? That's what the fascicles are. So when you are slicing that meat, you slice it against the grain, and that breaks up the connective tissue and makes it much more tender when you're eating it. Or maybe if you don't want to spend the money on an expensive tri-tip, maybe instead you get that pork butt and you put it in the slow cooker and you pour a beer over the top and you put the top on it and you put it on low for 12 hours. And when you come back 12 hours later, right, and you grab that meat, does it come out as one big chunk? No, it falls apart. However, when it falls apart, does it fall apart in just like puzzle piece size, irregular shaped pieces?
No.
No, it comes out as threads. Those threads are the fascicles. Because again, that low, slow cooking breaks down the collagen in that perimysium so that the individual fascicles, those threads, come apart. Like I said, we can debate whether or not meat is murder, but meat is definitely muscle. And these are some prime examples of that arrangement of muscle that, you know, we deal with on a weekly basis. So you may not have used these terms before, but you understand how this meat is arranged because we've done this before. All right. Excellent. So we have these individual fascicles, and then these individual fascicles, along with blood vessels and nerves, are then going to be bundled together by yet another dense irregular connective tissue. This dense irregular connective tissue, oops, that wasn't supposed to happen. My mouse ran out of space. There we go. Okay, start over here.
Epimesium.
I'm sorry?
Epimesium.
Yep, we're getting to that. Absolutely. So we have yet one more dense irregular connective tissue that wraps around the entire structure. Nope, not the sarcolemma. We'll get to that. So we have this entire structure that wraps around, and it again is a dense irregular connective tissue. So this structure that is being formed here is now the muscle, right? The organ. So this would be the bicep brachia or the tricep brachia or the rectus femoris or whatever it is. It is formed by, again, a dense, irregular connective tissue. That, based on location, as you guys have mentioned, is an epimesium. And this is how our muscle is arranged. This is how those muscle cells are held in arrangement so that they can be most efficient in doing their job. Now, there's one more cool thing that happens along with this. These connective tissues run the entire length of all the muscle cells. However, what happens is the collagen fibers from them can extend beyond. So for instance, the collagen fibers from our endomysium will extend out beyond the muscle cell. The collagen fibers from our perimysium will extend out beyond the muscle cells. And the collagen fibers of the epimesium will extend beyond the muscle cells as well. So notice over here, oops, all of the collagen fibers from all of these connective tissues extend out beyond the muscle cell. And notice as they extend out beyond the muscle cell, all the collagen fibers, oops, become parallel. And when we have all of these parallel collagen fibers, this basically becomes a dense, regular connective tissue. And what would the structure that would be formed by this dense, regular connective tissue be? No, not fascia is a good guess, but notice these are all extending.
Sarcomere.
Nope, not the sarcomere. We're going to get to that too. No, these are collagen fibers, a dense, regular connective tissue.
Muscle fibria.
Close. Dense, regular connective tissue. All the collagen fibers are aligned, and maybe this may attach the muscle to something, like a bone, since it's skeletal muscle, and a dense, irregular connective tissue that attaches the muscle to the bone would be indeed a tendon. Excellent. So the tendon actually is formed by the extension of these collagen fibers. And notice we see that in the previous picture. If we go back from the picture to the textbook now, we can actually see these things that we were just talking about. Notice here, we have individual muscle cells. So muscle cell, muscle cell, muscle cell, muscle cell, muscle cell, muscle cell, muscle cell. And notice around each muscle cell is a bit of connective tissue. This bit of connective tissue, yeah, I can go back in a second, Allison, hold on. This bit of connective tissue right here is an areolar connective tissue surrounding each individual muscle cell. And that is forming, based on its location, that is our endomysium. Then we have, it's not useful, let's use green, lighter green. Then we have this dense, irregular connective tissue that wraps around the whole thing. And that is our fascicle, that structure. And that is formed by the perimysium. And notice here's another fascicle here, and another fascicle here, and another fascicle here. Notice it's forming the grain of that meat that we talked about. And notice all these fascicles, plus blood vessels and nerves, which aren't on this picture, but they would be here as well, are all wrapped into the organ that is the muscle by our epimesium, which is also a dense, irregular connective tissue. And lastly, notice that all the collagen fibers coming out from it all form that tendon, which attaches the muscle to the bone. We can see this a couple more ways as well. Allison, I haven't forgotten what you asked for. I will go back to the picture, but let me show you the rest of these pictures because they're a little prettier than the ones that I drew. So notice here we have a cross section through the skeletal muscle, the organ, like the bicep brachia. Notice again, clearly, we see that there is a connective tissue that wraps around the entire structure That connective tissue is a dense, irregular connective tissue that, based on its location, we call an epimesium. Its job is to bundle all the fascicles together with the blood vessels and the nerves. So basically it forms the muscle. The same way the walls on the outside of your house form your house, this connective tissue on the outside forms the muscle. But we can grab one of these fascicles and pull it out. And if we grab one of these fascicles and pull them out, we can see that this particular fascicle contains 1, 2, 3, 4, 5, 6, 7, 8 individual muscle cells. And this fascicle is formed by a dense irregular connective tissue that wraps around these muscle cells that we call, based on its location, a perimysium. So notice both the epimesium and the perimysium are both dense irregular connective tissues. It's the same type of tissue. The only difference is their function. One wraps up the fascicles and one forms the fascicles. So the one that forms the fascicles we call a perimysium. The one that bundles all the fascicles together we call an epimesium. Now again, we can pull one of these 8 muscle cells out. And if we pull one of these 8 muscle cells out, we see that it is surrounded by a connective tissue, in this case, an areolar connective tissue. And this areolar connective tissue that wraps around the individual muscle cell is an endomysium. So this one's the areolar connective tissue, and it forms the endomysium. All of these names are the names based on location. And notice if we look inside the muscle cell, there's a whole lot of stuff going on inside here as well. So we still need to go even further, break it down even more to make sure that it makes sense. But here we see this really nicely. And let's truly beat the dead horse. We can see this histologically as well.
Excuse me, sir. These
Yes.
slides, do we have these slides in the handout?
Yes, these slides should be in the handout. That is correct. And I think all of these pictures, I think, are from your textbook as well. All right. Here we have a histology slide. This one might not be on your handout, but again, I found this with the almighty Google. Oh, that reminds me of something I wanted to show you real quick. But let's take a look at it. Notice here, we see an individual muscle cell. And here's an individual muscle cell. And notice there's a little bit of white in between them. That little bit of white in between them, what tissue type would that be? I don't think the garbage truck knows what it is.
Dense irregular.
No, around each individual muscle cell, it's which one?
Areolar.
Areolar, excellent. And what would we call it based on its location?
Endomysium.
Endomysium, excellent. Here we see a connective tissue wrapping around a handful of muscle cells. What do we call this structure?
Fascicle.
Fascicle, excellent. What type of connective tissue forms the fascicle?
Dense irregular.
And dense irregular. And what do we call this dense irregular based on its location?
Perimysium.
Perimysium. Excellent. And lastly, we have one more connective tissue that is forming around the whole thing, bundling all of these fascicles together, as well as we see a blood vessel in here as well. What type of connective tissue is this that wraps around and forms the entire muscle?
Regular connective tissue.
Dense irregular connective tissue. And what do we call it based on its location?
Epimesium.
Epimesium. Excellent. We see this histologically. And then again, we have all these great illustrations that show it as well. Again, we have a single muscle cell with that areolar connective tissue wrapped around it, that endomysium. We have our perimysium forming the fascicles, right? And again, this one, 3, 6, 7 fascicles in this one, formed by that dense irregular connective tissue called the perimysium. And all these fascicles and blood vessels and nerves are bound by the epimesium, a dense irregular connective tissue. And then
Sir, if you go back one slide, go back. So I don't see that slide. I don't know why in the.
Well, as I said, I mentioned the histology slide might not have been.
No, I'm not talking about this one. It's here. I can see it.
Okay,
well, this one.
Okay. Like I said, I made these outlines at the beginning of the semester. I do tinker with these. That should be somewhere in there, but if not, this picture is in your textbook. So these material is from your textbook. And worst case scenario, you do have the recorded lecture. You can go back and see these things again as well. All right. Last thing, remember, we talked about a tendon is what connects a muscle to bone. And I think we use this term, but I want to remind you about it again because we're going to be using it in this section. Some tendons can become a big, broad, flat tendon. For instance, we have a big, broad, flat tendon that goes over the top of our skull, or a big, broad, flat tendon that goes over our abdominal muscles. A big, broad, flat tendon we call an aponeurosis, IS singular, aponeuroses, ES is the plural of that. So lots of different ways of seeing this individual muscle cell. And then now, again, we go back to the pretty picture here. You see that arrangement as well. And I can save that and always put that on the website. Although I think the pictures from the textbook do a better job of showing this. All right. Questions on that? Again, if this isn't making sense now, you need to let me know now because this is only going to get worse.
Sir, again, my question goes to the same slide. I can see this slide here, the slide of the histology.
Okay.
But I don't see the slide. You explain all the epimesium, perimysium, the previous slide. I don't see it here. I don't know why.
Because it looks like the histology slide is over the top of it. Now that I'm looking at the slide, the histology image is over the top of it. Again, many of the slides are layered with information, and unfortunately only the superficial information comes up. But remember, these slides are just an outline to help you with this material. Most of these pictures or something equivalent to it are in your textbook. The goal isn't to memorize these pictures. The goal is to make sure that you understand the information. And so whether it's that picture or this picture or something like it, I guarantee your textbook has something like that, or like this one here, which is showing the same thing, and also showing how we are going to further break this down. Because so far, notice we've only done the left side of this, and we still have more breaking down to go.
I don't mean that I'm going to memorize the picture. So what I'm saying is this is a missing slide or a PowerPoint slide. So what I'm saying in the slide so that I cannot see it, this is the only question I have.
Okay. And I apologize for that, but what you need to understand is that when I make these slide outlines, I am just taking the copy of the slides from the presentation. The slides are dynamic, meaning that there are layers of information that are being presented on it. In this particular case with this slide, that histology slide is layered on top of the pictures that are underneath. And so that when I take that slide outline, that when I print it, it just shows the superficial pieces of information. So again, I think it is important that you have these resources as a way to follow along and to help you to understand the material. But again, the outline is not the equivalent of the lecture. So while I appreciate that not everything is not going to necessarily be on those outlines, that is the point of an outline. An outline isn't supposed to be the script of what I'm saying. It is something to help to direct you towards the information that you're responsible for. Is this slide on your outline?
Yes.
This slide has the exact same information as that previous one. Notice here, we see that epimesium wrapping around the follicles, the fascicles, and the blood vessels and nerves. We pull out a fascicle. We have the dense irregular connective tissue forming the fascicle, forming the perimysium, wrapped around the muscle cells. And then here we pull out a muscle cell, and we have the areolar connective tissue wrapped around it. So again, there are multiple ways to see this information. And if one of those isn't viewable because of the dynamics, the problem is if I tried to show every layer to every slide, those lecture slides would be 3 times the length that they already are. So just again, it's a tool to be used, but it's not the exclusive tool that you should be using to be successful in this class.
Thank you,
All
sir.
right.
Thank you.
Yep, no worries. I appreciate your concern with that. Thank you. All right, so we are now down to the level of a single muscle cell or muscle fiber. Remember, we can use those terms interchangeably. But as we can see, there is all sorts of hunk and junk going on inside of our muscle cell. And so what we need to do is we need to now further look at and break down the anatomy of a muscle cell, starting with something unique, how we make a skeletal muscle cell. Up to this time, we've been talking about epithelial cells and we've been talking about connective tissues. And remember, with connective tissues, there was the mesenchymal stem cell that is able to divide to produce any of our connective tissue cells. What we have here is a different type of embryonic cell, and that embryonic cell is what is called a mesodermal cells. These mesodermal cells are embryonic, pluripotent. Wow, that's huge. Let's hold on, change that. I don't like that either. These are embryonic, pluripotent stem cells that, as an embryo, divide to form myoblasts. Myoblasts are, of course, the immature cells. And these immature cells are what form our muscle cell. But it isn't one myoblast that becomes one muscle cell. Instead, as we can see from this illustration, hundreds of these myoblasts will fuse together. And when they fuse together, they form that single skeletal muscle cell. This explains a couple things. One of the things it explains is why there are multinuclei, because it is hundreds of cells coming together, bringing their nuclei with it. So we can get a cell that could have as many as 100 nuclei inside of them. That does bring us to one question: why? Why do we need all those nuclei? What does having multiple nuclei do for us?
In the nuclei, we have DNA and we have protein.
Absolutely, DNA, and DNA is the instructions for making proteins. Remember, we said muscle cells are going to be chock filled with perfectly aligned proteins. So having multiple nuclei is going to help a lot with protein synthesis. Right? However, Dina brings up a good point. Right? Normally, when we talk about regeneration, we know that a cell has to be able to divide to make new cells, right? And that's normally how we regenerate. However, if we have this skeletal muscle cell that is comprised of hundreds of muscle cells that have fused together, is this the kind of thing that can easily divide and become new cells? No. So remember, as we talked about, regeneration is very limited. And the reason for that is because this is a very complex cells. But remember, there can be some regeneration. And the reason for that happens to be this cell right here. Notice when our muscle cell formed, most of the myoblasts fused together to form the skeletal muscle cell. But most is not all. There are a few leftover myoblasts. And these few leftover myoblasts become cells that are called satellite cells. These satellite cells are immature myoblasts that, if the muscle cell is damaged, they are able to divide, make new myoblasts, and those new myoblasts can either fuse with the muscle cell to repair the muscle cell or actually replace the muscle cell if it has been damaged. However, do we have an unlimited number of these satellite cells?
No.
No, we just have a few of them. So that's why we can heal, we can regenerate minor injuries to the muscle cell with these satellite cells. But if it's a large injury where a large portion of muscle is lost, there's not going to be enough satellite cells to be able to replace that. And so that's why the fibrosis, the scar tissue, forms instead. So it is these satellite cells that allow for the limited regeneration. There's one other implication to this as well, right? Here is a current muscle cell inside of your arm right now, right? As I mentioned, I've been grading the lab exams from the bones, and so far it hasn't been spectacular looking. So as a result of that, you, like me, may be exhausted with this class, and we decided to just chuck it. Let's forget all of this. I did say chuck it. And we will, instead of meeting to talk about anatomy and physiology for 4.5 hours, we're going to get together and work out together, lifting weights on camera with each other 4.5 hours a day, right? If we were to do that for the next 8 weeks, all get together for 4 hours every day for 5 straight days and work out, are our muscles going to be bigger as a result of that?
No.
No? You work out 4 hours a day for 5 days a week for 8 weeks, you're not going to have bigger muscles?
Why not?
Yeah, your muscles are absolutely going to be bigger as a result of that, right? Our muscles were going to be all big and strapped and strong, right? And the reason for that, our muscle gets bigger not because we make more muscle cells. Instead, what happens is we make more proteins. We don't make more muscles, we make more proteins. So what happens is that same muscle cell we fill with more proteins. So what happens is we make more proteins, and as a result of making more proteins, the muscle gets bigger. The muscle gets bigger and stronger. Notice we're not increasing the number of muscle cells, our cells themselves get bigger. This type of growth is what we call hypertrophy. Right? So our muscles get bigger thanks to hypertrophy. Now, of course, final's going to come around. If we've been exercising for 4 hours a day, 5 days a week, we haven't been studying anatomy and physiology, which means we're going to fail the final, which means we have to take this class again next semester. And as we're taking this class again next semester, that means we're not working out anymore and we stop using those muscles. Or worst yet, you break your arm and your arm gets put in a cast. And 6 weeks later, they take the cast off and you hold your two forearms up to each other. And what do you notice about the size of the forearm that was in that cast?
Atrophy, so it goes smaller.
Exactly. It gets much, much smaller because, as I mentioned, our muscles are very dynamic with all these proteins. And so what happens is if we don't use the muscle, exactly, that's what's happening to you right now. If you don't use the muscle, then what happens to the muscle is those proteins get broken down.
Atrophic.
Exactly. Broken down with not non-use, let's say it that way, with non-use, and that type of loss of muscle mass, as you guys pointed out, is called atrophy. So the muscle atrophies as a result of it. So our muscles change size, not by adding or subtracting muscle cells, but by making the muscle cells bigger or making the muscle cells smaller. Now, one of the things you're going to be responsible for on this exam is actually being able to look at bodybuilders and recognize the muscles. Because some of these bodybuilders, like the one on the very first page that we looked at, the muscles get very large. They dehydrate their body so that the skin just slaps on the surface of the muscles, and we can easily identify the muscles by looking at them. With that kind of insane muscle mass increase, is it possible that if you're really working out 6 to 8 hours a day, 7 days a week for years and years and years, that you may be able to make some new muscle cells doing that? Absolutely. And that type of growth would be what we call hyperplasia. But is that the normal way that we grow muscle cells? No, absolutely not. So again, primarily when we expand our muscles, we grow via hypertrophy and typically not hyperplasia. Hyperplasia would be adding new cells. Again, some of these monster workout people do get some hyperplasia that can occur from that extreme bodybuilding, but it takes that type of extreme exertion to cause that to happen.
So we can say that the hyperplasia is an abnormal growth of the muscle cells, right?
So I wouldn't say abnormal. It isn't like there's something wrong when that happens, but it takes extreme stress and exertion to cause it to occur. So it is not the common way that our muscles increase in mass. Yes, Netflix and Cheetos would help those people. Right? But, part of it's, that we're all locked up at home, as Dave talked about, right? Due to the corona, we're all going to come out of the corona one of three ways. You're either going to be a hunk, you're going to be a chunk, or you're going to be a drunk, right? And if you work really hard, you can be 2 out of 3 of those. All right. Questions on that? All right, excellent. So now we need to talk about the muscle cell, and we need to talk about the anatomy of the muscle cell. The good news is we have a good starting point. We know the anatomy of the muscle, of a cell. However, a muscle cell is very specialized, and because it's very specialized, are things going to get special names? Absolutely.
Yes.
So let's take a look, and let's do this again for ourselves. Allison, did I ever come back to this long enough for you to get what you needed from this?
Yes, thank you.
Okay, excellent. All right, I think I saved this. I'll save it one more time just to make sure, and then I'm going to go ahead and clear this. So let's talk about a muscle cell. We know that muscle cell is going to have a plasma membrane on the outside. So let's put that, and then we will cheat and do that there. Excellent. That's a good starting point. Although I'm going to cheat one more way. Perfect. Excellent. So we have this long cylinder-shaped muscle cell, and like all cells, we know it has a plasma membrane on its outer surface. And now this I'm going to need to make smaller because we're going to need lots of space. That's probably decent. A plasma membrane. However, the plasma membrane of our muscle cell is specialized. It is specialized to produce and propagate a muscle action potential. What's it mean to propagate?
Create more of.
Not a bad guess. Propagate, we do can mean to produce more of, things along those lines. In this case, what we mean by propagate is to spread, right? Again, I'm locked at home in quarantine, right? Married, 2 daughters that are teenagers, 2 dogs that are female, right? I'm swimming in the estrogen ocean here. So like most mornings, I wake up and I grab my yardstick, my wooden yardstick that has that nice metal end of it, and just to try to get through the day, I shove that into the light socket, right? Does anything happen when I do that? If I were to take a wooden yardstick and shove it into the light socket, anything going to happen to me because of that?
No.
Yeah, exactly. I'm sitting here. So no, of course, nothing happened. Absolutely. However, if I had one of those metal yardsticks and I stuck it into a light socket, would anything happen then? Yeah, because it's able to conduct. And that's really what propagate means, to conduct from one end to another, right? With that wooden yardstick, even if it has a metal end, I may get a shock when I put it in. I mean, it may shock, it may spark, but it's not going to travel up the wood. The wood can't conduct it. So in this case, what happens is we produce an action potential and it spreads down the entire plasma membrane. Makes the plasma membrane very special, so we give it a special name. The plasma membrane of a muscle cell we call a sarcolemma. So the sarcolemma is the plasma membrane of our muscle cell. Of course, we also know the stuff inside of the cell is the cytoplasm, A muscle cell has all the normal stuff we would expect to have inside of a cell. Ions, more sodium, right? More potassium, less sodium, things along those lines on the inside of the cell. But remember, we also talked about cells having inclusions. Inclusions are special molecules or special materials that there are a lot of inside of this cell. One of the things that skeletal muscle cells have a very large amount of is glycogen. What is glycogen again?
Stored sugar.
Yeah, it's stored sugar, absolutely. Why might our muscle cell want a massive amount of stored sugar inside of it? Energy, absolutely, to produce ATP. We need to be able to produce that ATP to move that muscle through space. Of course, to do that efficiently, we also, where am I? This muscle cell inside of it has a very special protein called myoglobin. Anybody know what myoglobin does?
Stores oxygen.
Stores oxygen, absolutely, right? If you think about it, it sounds very much like hemoglobin, which is what stores oxygen inside of our red blood cells. So that myoglobin stores oxygen. Because if we are going to efficiently break down our sugar, we need oxygen to completely break it down. That makes that breakdown as efficient as possible and gives us the most ATP. Of course, if I'm going to use oxygen to completely break down sugar, wasn't there an organelle that really helped to make that process efficient?
Mitochondria.
Exactly. We are also going to have a massive amount of mitochondria. So there's all these specializations in the cytoplasm, and since there's specializations in the cytoplasm, not surprisingly, we give the cytoplasm a special name, sarcoplasm. So our sarcoplasm is that cytoplasm on the inside of the muscle cell that, again, has a massive amount of glycogen, has a massive amount of myoglobin, has a massive amount of mitochondria. All right, so far so good? Excellent. Let's go back to our sarcolemma then, because the sarcolemma of our muscle cell does something interesting. Yes, the sarcolemma is the plasma membrane on the outer surface of the cell, but there are invaginations, there are folds of our sarcolemma that penetrate deep down into the muscle cells. These invaginations of the sarcolemma are what are known as the transverse tubules. And the good news is on a lecture exam, when you're writing an essay, once you've written out transverse tubules once, you can abbreviate it to T-tubule. Now, these are extensions of the plasma membrane, basically a fluid-filled tunnel. And this fluid-filled tunnel is going to contain extracellular fluid or interstitial fluid. So like the fluid that surrounds the muscle cell, it has a high level of sodium, a low level of potassium, a high level of calcium, and so on and so forth.
I'm sorry, it's an extension of what?
The plasma membrane, the sarcolemma. So it is a fold in the plasma membrane that folds down deep into the muscle cell. All right. So far, so good. All right. There are some other organelles inside, some that we are familiar with, starting first with, and I may have to decrease the size of this once more, are smooth endoplasmic reticulum. However, if you remember the function of the smooth endoplasmic reticulum, we talked about how it can be used to make lipids. It has some minor detoxification. But remember, we also said it could be specialized for storage. And that's what it does here. Here in our muscle cell, it is specialized for the storage of calcium. Massive, massive amounts of calcium. In fact, outside of the matrix of bone, the smooth endoplasmic reticulum of a skeletal muscle cell has the highest concentration of calcium in the body, right? Second overall. First is the bone matrix. Second is the smooth muscle of the endoplasmic reticulum. The smooth endoplasmic reticulum of our skeletal muscle, which of course means it has to have a special name, and it is sarcoplasmic reticulum. So let's draw a little of this. We'll use yellow for this. I'm going to draw it as a narrow tube, but as we'll see, it's kind of this weird mesh-like structure. However, what's interesting about the sarcoplasmic reticulum is when the sarcoplasmic reticulum gets close to the transverse tubule, it enlarges. And this enlargement of the sarcoplasmic reticulum is referred to as the terminal cistern. The terminal cisterna. Notice sarcoplasmic reticulum buds up against the transverse tubule on the left here, but it also buds up against the transverse tubule on the right side as well. So we have more sarcoplasmic reticulum and a second terminal cisternae. So notice here, and I will use that, and I will use that, and I will use that. Notice what we have here are kind of three structures together. We have two terminal cisternae, plus the T-tubule. And these three things together are called the triad. So these three things together are what we refer to as the triad. The two terminal cisternae and the transverse tubule. All right. Questions on that? All right, I'm going to cheat and draw a second sarcoplasmic reticulum. Oops, no, needed to be yellow again.
Sir, can you go back and just said how the terminal cisternae are formed?
They are folds in the plasma membrane. So the plasma membrane goes along the surface and then folds in here. Again, I appreciate that my art isn't the greatest. So let's cheat and go back to the lecture slide. Notice here from the lecture slide, and we, I think, see it best over here in the partial cross section. Here we have the plasma membrane that goes along the entire surface and circumference of the cell. But notice there are folds in that plasma membrane that penetrate deep down, and these fluid-filled tunnels that penetrate deep down into the muscle cell are known as the transverse tubules. So they're just folds of the plasma membrane that penetrate deep down into the muscle cell. Notice here in blue is my mesh-like sarcoplasmic reticulum. Here are the enlargements, the terminal cisternae, and notice the two terminal cisternae and the transverse tubule. These three things here are my triad. So if you don't like my art, like I said, your book's got very pretty pictures that show this nicely. However, I want you to see one more thing. Notice the sarcoplasmic reticulums and the transverse tubules don't just penetrate as a straight deep line. They wrap around all these special thread-like organelles. These thread-like organelles are the most common organelle in our skeletal muscle cell, and it is a very specialized organelle known as a myofibril. So let's cheat and go back to my picture and draw one of these. We will do it in green. There are these long thread-like organelles that run the entire length of the muscle cell. And these thread-like organelles are called the myofibrils. The myofibril is just chock filled with proteins. And of course, these massive amounts of proteins that form them are going to have a special name. We are going to call them myofilaments. Great question. The terminal cisternae are just the enlarged ends of the sarcoplasmic reticulum. So the whole organelle is the sarcoplasmic reticulum. The enlarged ends are the terminal cisternae. Now, these myofilaments are precisely arranged. In the myofibril. Oops. Forming a structural and organizing structure a repetitive, let's say it that way. And that repetitive organizing structure is called a sarcomere. These sarcomeres are the structural and functional units of the myofibril. I always kind of think of this as like if you're ever stuck at a train crossing and you see the train go by, and that one train is made up of 85 grain cars just going by. And it's that exact same grain car that you see again and again and again and again. That's exactly what's happening here. This myofibril is made up of a sarcomere, which is connected to the next sarcomere, which is connected to the next sarcomere, which is connected to the next sarcomere, and so on and so forth. That repetitive pattern of proteins is what is known as the sarcomere. And that sarcomere is the structural and functional unit of the myofibril. By functional, obviously, I mean it is what is going to cause the contraction. But structurally, it is the precise arrangement of the proteins in this myofibril, in this sarcomere, that gives the muscle its stripes. So this both produces the tension, produces the force that is going to move the muscle, but it also, like I said, is what the precise arrangement produces the stripes. So the striations, those stripes of our skeletal muscle are formed by these sarcomeres as well. Again, I fully appreciate my art is not the greatest for this. So let's take a look at the pretty pictures from your textbook. This is a good way to re-emphasize the material and the vocabulary.
Is sarcomere the name of the protein or just the arrangement of the protein?
It is the structure that forms, it is the arrangement of the proteins. That sarcomere is made up of over a dozen different proteins. I'm going to hold you responsible for the names of 7 of them. But know that there are over 20 of these special proteins found in our muscle cell that help to provide the precise arrangement and function and structure of these. We're going to learn the most important ones, but there are other ones to be found in here as well. All right, so let's go through the vocabulary that we've done before so that we can even further break it down. The plasma membrane, that specialized plasma membrane that allows us to produce and spread, right? If you don't want to use propagate, we can just say spread. Produce and spread the action potential is the sarcolemma. And all the hunk and junk inside is the sarcoplasm. Like we talked about, there's going to be a large number of glycogen granules, large amount of that special protein myoglobin. So we have our sugar, we have something to store oxygen, and we have those massive number of mitochondria to efficiently make ATP. There are specialized folds or invaginations of our plasma membrane called the transverse tubules that penetrate all the way down and through our muscle cell. They're fluid-filled tubes that contain interstitial fluid. So what's inside the tubes is different from what's inside the muscle cell. Associated with those transverse tubules is our Sarcoplasmic Reticulum, our smooth ER. That's shown here in blue. It stores a massive amount of calcium. As we know, calcium makes cells do wonky things, and contracting is definitely a wonky thing. So that ability to contract is thanks to all this calcium that is stored in our sarcoplasmic reticulum. So this whole blue organelle is the sarcoplasmic reticulum. However, the enlarged end of it is that terminal cisterna. Right, so the 2 terminal cisternae and the transverse tubule in between, these 3 structures together form our triad. As you see, the sarcoplasmic reticulum, the transverse tubules completely surround our myofibrils. Our myofibrils are the primary organelle of a skeletal muscle cell. They're these long thread-like organelles that run the entire length of our muscle cell. And there are contractile organelles. They are what produce our force, generate our force for contracting the muscle. It is comprised of massive numbers of proteins called myofilaments. Some of them, as we'll see, are thick and thin, so they're sometimes specialized that way, although we're going to distinguish them into 3 main categories. And they form a repetitive pattern called a sarcomere. We haven't talked about the anatomy yet, but I will cheat. If you look closely, this right here is one sarcomere. So from this zigzaggy point here to this zigzaggy point here, that inside of that is one sarcomere. And so basically our myofibril is made up of hundreds of repetitive patterns of this sarcomere. And that's what we need to do next. We need to now pull this myofibril out, look at this sarcomere, look at the proteins that make it, and see how they're arranged. So here, notice again, from one zigzaggy line to the other zigzaggy line, I have a single sarcomere. And a myofibril is going to be made up of dozens, if not hundreds of these. They're perfectly and precisely arranged proteins, both thin and thick protein filaments, collectively known as the myofilaments, that give this precise arrangement. And notice here, we actually have a view of a myofibril with an electron microscope. Using an electron microscope, we can actually see the individual proteins inside. And notice there's one area where they're very, very dense. That forms the dark stripe. And then there's one area where they're very, very sparse, and that forms the light. So notice dark, light, dark, light. It is the precise arrangement of these proteins that give our skeletal muscle and also our cardiac muscle their stripes. So this is how the tigers get their stripes, this precise arrangement of these proteins. All right. Of course, we have to identify what these proteins are, but that again takes us with a further deeper dive into this material. So before we do that, any questions on that? All right, we're ready for our next break. Before we do this, I want to do one more thing real quickly before I get too far away from the material. If you remember, one of the things that I mentioned is that one of the preparations you are responsible for on this exam is what they call a teased smooth muscle prep. Remember, the smooth muscle is normally arranged in layers, but literally what they do is they take a couple needles and they tease apart the muscle cells. And when you do that with smooth muscle, this is what you end up with. Now, obviously, if you have a chunk of tissue and you're using a couple needles to pull them apart, are you going to damage some of the cells doing that? Absolutely. So we see plenty of cell pieces here. But the other thing that we see here really nicely is that nice arrangement. Why are you not letting me? There we go. That doesn't. So for instance, here we can see a portion of a cell, long shape, centrally located cell body. And then notice you can see that it's cut off here. But you can see plenty of other examples of that nice spindle shape. Big, large, centrally located body and two tapering ends coming off. Right, single centrally located nucleus and like long, narrow thread-like structures coming off. So we can see this. When you look at this under the microscope, it should be something that's very easy to recognize. So of course, one of the questions I could ask is to identify this tissue type, identify this prep. And of course, you know, this is smooth muscle that has been teased. But anytime I show you histology or a model of one of these tissue types, you also are going to be responsible for its characteristics. So for instance, I could say, identify two characteristics of this muscle tissue. So remind me again, some of the characteristics we know about smooth muscle.
So they're spindle shape.
Spindle shape, excellent. What else?
So unicells, one cell.
Uninuclear, excellent.
Involuntary.
Involuntary, excellent.
Non-striated.
Non-striated, excellent. Right. Give me at least one more.
Generation. Ability.
Highest rate of regeneration of the muscle tissues. Where do you find it?
In hollow tubes, wall of the hollow tubes.
Found in the walls of hollow organs, exactly. So I could show you something like this, or I could show you something like this. Notice, for instance, in your modules, as you've hopefully come down, you'll see that there are some muscle classroom models. So notice I could give you a picture of one of these models, like let's say, for instance, what muscle type do you think that is? Muscle of what type? A model of what type of muscle?
Skeletal muscle.
Skeletal, absolutely. Cylinder shape, multiple nuclei, striations, right? This is skeletal muscle. So I could ask you to identify the tissue type, or I could ask you for characteristics. What are some of the characteristics of skeletal muscle?
So they are
multinucleated,
voluntary,
striated.
They're mostly found on the skeleton.
Most
Of the
are
body.
connected to bones, absolutely. Things along those lines. Limited regeneration, excellent. What's this one?
Smooth muscle.
Smooth. We see that spindle shape. We did that. Oh, it doesn't really show it on this model. Does the other one show better? No. All right, I'll cheat. In this muscle, it's three-dimensional, but you can see a teeny bit of it here. Notice there's this little bit of gray material on the outside. If you were to rotate this model, what you would see is there is a sheet of this gray material like this that kind of wraps around the outside of it like that. Based on its location, what type of tissue would that be based on its location? What do you think this gray stuff in between the individual muscle cells wrapping around it would be? Absolutely, it'd be the endomysium. And so what tissue type would that be?
Areolar connective tissue.
Areolar connective tissue. Excellent. Perfect. And then, of course, what do we have here?
This is
Cardiac muscle. Excellent. We see stripes, centrally located nucleus, branching shape, right? These black lines, which we'll talk about in a little bit. But so we have things like this. So we have these models. You need to know the characteristics of the individual tissue types. I have some other models. These are great. And when I say something is great, that should be a huge warning that this is easily something that could be on the exam. This here is a model that is a representation of a portion of a single muscle cell. So with this single muscle cell, notice this tissue here on the outside would be our areolar connective tissue, would be the endomysium, whereas this right here would be the plasma membrane, or more specifically the sarcolemma. If this is the sarcolemma, and this here is the endomysium, then does anybody have an idea of what this blob right here might be? Notice it's right up against the muscle cell. It's not inside the plasma membrane, so it's not a part of the muscle cell, but it's actually located inside the endomysium, butted right up against the muscle cell. Alina's gets the prize. Absolutely, this is that satellite cell. Remember we talked about how those immature myoblasts are right there next to the muscle cell. So if that muscle cell gets damaged, it can divide to replace it. Notice, and again, if we zoom in a little bit, that's pretty. I don't know if I can grab that. Hold on, I gotta do this. Oops, wrong button. There we go. Center it this way. Perfect. Notice here in orange would be our transverse tubule. Notice here in white is our Sarcoplasmic Reticulum. Here would be our enlargement, would be our terminal cisternae. And then these three things together would be our triad. Notice here we see the myofibrils and many of the proteins that are making them up. And we can see this from multiple points of view, looking straight down from the top, looking from the side. We'll talk about what that is in just a minute. But notice also there is a second classroom model. Notice this one doesn't have a satellite cell, but it does have the areolar connective tissue, the plasma membrane, the transverse tubules, the sarcoplasmic reticulum, the triad, the myofibrils, mitochondria, right? Or, pardon me, that's the nucleus. These are the mitochondria, and so on and so forth. So these models are great examples of the single skeletal muscle cell anatomy that we've just talked about, and I guarantee they're going to be on the exam. We also, as well, in a couple of views of that, we also have the model of the sarcomere that we'll look at as well. So there's some great resources here online in your modules that you should be using to take advantage of and to look at these things as we're doing that, all right? We'll save this sarcomere model till we finish our lecture because we have to identify these proteins and talk about them. But like I said, at the end of lecture today, we have a lot of resources that if you're not already using, first and foremost, shame on you. But secondly, definitely let's take a look at them to see what you can do to help yourself to be successful on this. All right, we have a lot more anatomy to go with these sarcomeres. So before we do that, let's come back fresh. Looks like it is 10:35, so that means coming back at 10:50, and we will restart at 10:50. So
is there any way to label these models, like to put the labels on them and to better understand and to better to be more successful in the exam, label them and then remove the labels later on?
yeah, I mean, you can print. So what I've had students do in the past is, again, these are all pictures that you could print out for yourself, label them yourself so that you get the ability to do that. And you could do it one of two ways. I've had students who either laminate these, or the easiest thing to do is put it in a page protector. When you do it in a page protector, you can write then with a marker on the outside of it, label those things, and then erase it so that you can do it again. Or what I've had a lot of students do is label these things, and then you put Post-it notes over the top of them. So that way you can hide the answers and then look to see what the answer is. So there's lots of ways that you can use these resources to help you to be successful. Okay.
Thank you.
All right, so we will restart at 10:50 and I will start the recording at that part. Excellent. So I will see you guys in 15 minutes. All righty. More fun vocabulary. So our goal now is going to be to take this myofibril and break it down into its basic building blocks. Again, we know it is that sarcomere, which is that repetitive pattern. But that sarcomere is made up of dozens of proteins that are precisely arranged. And as I mentioned, I'm only going to hold you responsible for 7 of those. So let's talk about those 7 proteins. And again, I think it's easier sometimes to look at a more simplistic view of this. So let's go ahead and clear this. When we talk about we're talking about our myofibril. That again is comprised of sarcomeres. Which is formed by a collection of proteins. And again, button with vocabulary, myofibrils. Pardon me, myofilaments. Now, there are 3 main types of myofilaments. And so let's talk about and identify those 3 main types. The first of those 3 main types are our contractile proteins. And of those, there are 2. Anybody know what the 2 contractile proteins are? I think someone mentioned them earlier, but we'll go ahead and ask again. Myosin and actin, excellent.
Using actin and
Yep, myosin and actin, excellent. Myosin, okay, both of these, let's do this over, where are we going to do this? We'll do this here. Both of these are dimers. Anyone remember what a dimer is again? A dimer is comprised of 2 protein subunits. So let's talk about myosin first. Myosin is kind of shaped a little bit like a golf club. It has a long, I don't like that. It has a long thread-like tail. And then off of it, it has a large head region. So it has a tail and it has a head. And it is a dimer. So basically what happens is 2 of these things, the tails intertwine, and then you have the heads coming off this way. So that is what a single myosin subunit looks like. 2 tails intertwined with 2 heads. And the heads are, again, that fancy word we mentioned earlier, a motor. What does a motor do again?
Converts energy.
Yeah, converts chemical energy into mechanical energy. So in this case, that chemical energy is, of course, ATP, into a mechanical energy. And that mechanical energy in this case is going to be a pivot. Basically, what happens when our myosin head uses the energy for the ATP, from the ATP, it basically pivots forward. So basically it rotates. So again, it grabs and then it pivots, right? It pulls. That's what it does. It grabs and pulls, right? Grabs and pulls. Notice I'm grabbing and pulling here right now. Why am I not off the screen? Why am I not moving anywhere as I'm grabbing and pulling? Aside from the fact that I'm an amazing mime, how about the fact that I'm not actually grabbing onto anything? Exactly. You can't climb a ladder if that ladder doesn't have rungs. You need something to grab onto. And that's what the actin is. Actin is also a dimer, but it is a globular protein. So it's these two ball-shaped dimers that are kind of stuck together like this. And on them, there is a small little binding site for the myosin. So this actin has handholds on it so that the myosin can grab and pull. If you remember, actin forms those actin filaments that we talked about in the cytoskeletal structure. So what happens is these dimers form a big long line, all bound together. And all of them have the binding sites on it. And so basically it forms this rope that the myosin head can grab and pull on. Now I'm going to cheat here and erase all of that so that we can kind of draw what this arrangement looks like. What we have, and we'll start here with, well, I guess we'll use red. Oops, I want that there like that. You get a big, huge, thick myosin filament. And that myosin filament is what's called the thick filament because all those tails come together. Hundreds upon hundreds of tails all wrapped together into this big, large filament that then has all the head regions sticking out from them. And we can go ahead and put some heads on these. So at the center of our sarcomere, we have this big, huge, thick filament made of myosin, made up of all these tails and all these heads. So this is our thick filament made of myosin. All right. And let's make that a little smaller. There. That can work. Actually, let's cheat and do that. Perfect. So that's our thick filament, our myosin. And rather than drawing a bunch of ball shapes, I'm just going to draw it as a line. We have our actin arranged so that the myosin head can grab and pull on it. So this is our actin. I'm going to cheat and move it a little further away because it serves my purpose. But this one, obviously it has to be close enough so it can grab it. So this here, I need you over here. I need you over here. Aha. And this is our actin. So our actin and myosin are arranged in this fashion. And those are our two contractile proteins. They're the ones that are going to change the shape of our muscle cell, change the shape of our sarcomere. They're going to be the ones that generate the tension. Okay. Now, myosin has that head with the ATP wanting to grab and pull on the actin. And it wants to grab and pull on the actin all the time. But are your muscles in a constant state of contraction all the time? No. And the reason for that is we have regulatory proteins. Regulatory proteins are the proteins that are responsible for turning the contraction on and off. And there are two of these as well. The two of these are called troponin and tropomyosin. These are our two regulatory proteins. Tropomyosin is a long thread-like protein that will make green that basically sits on top of the actin, stopping the myosin from grabbing onto it. Again, I have to work on my analogies, but normally we're sitting inside of 411 inside of the classroom at American River College when I'm giving this. And I point out to you that on the way home, you may want to hit that McDonald's that down is off of Madison. And if you've ever been in the drive-through at that McDonald's at Madison, you will notice that right there next to the drive-through is a ladder that goes onto the roof of the McDonald's. And every time I'm there in that line, I want to climb that ladder to go up onto the roof. But I'm not able to. Does anybody know what the heck I'm talking about? Anybody remember that? I know it's been a while since you've been on campus, but what they actually have is this big, huge, thick piece of plexiglass that sits on top of that ladder. And because that plexiglass is in the way, you can't grab onto the rungs and you can't get to the roof of the McDonald's. And that's basically what the tropomyosin is. It's that big, huge piece of plexiglass that sits in the way, stopping you from being able to climb. Well, Allison, you're just not committed then, because if you're in that line seeing that ladder, you should want to go up to the top of it. But yeah, it's got this big, huge piece of plexiglass over the top that stops you from being able to climb. And that's what the tropomyosin is. It's this big, huge barrier that stops the myosin from grabbing onto the actin. And so that is our tropomyosin. Right? And that's great because it stops the myosin from interacting with the actin and the muscle stays relaxed. Except, there you go. Okay. All right. So you're excused then. Except eventually we're going to want the muscle to contract. And so if we want that muscle to contract, there's got to be a way to move the tropomyosin out of the way. And that's where the troponin comes in. Troponin is basically a handle-like protein that is found on the tropomyosin. So this is our troponin. And what that troponin is able to do is it is actually able to rotate the tropomyosin out of the way. So when it needs to, that troponin can actually rotate the tropomyosin so that it is out of the way. We need that handle on there where it's able to rotate it out of the way. And then the binding site is exposed. Myosin can grab onto the actin and the muscle is able to contract. So when we want the muscle to contract, we move the regulatory proteins out of the way. When we want the muscle to relax, we move the regulatory proteins in the way. So in this fashion, by moving the regulatory proteins into and out of the way, we are able to get the muscle to contract or relax. All right. Questions on that?
So
Is that why?
Oh, sorry, go ahead.
I was just going to ask if that's why people who are having cardiac events have high troponin levels, because it's not acting out right.
Great question. No, That's a great question. In this case, the troponin that you're looking for in that case is a different protein. It's a different version of the protein only found in cardiac muscle. So yes, when someone is having some type of cardiac issues or damage, that protein can be released out of the cells into the blood, but it is a slightly different version of the troponin. It's a different, it is a different type of troponin found in the cardiac muscle than found here in the skeletal muscle. Allison, was that your question as well?
Yes, it was.
Excellent. There you go. Like mine's. I like it. Excellent. Yep, it is absolutely one of those things you're looking for is troponin levels, but it's a different version of the troponin molecule. Excellent. But it is in muscle cells, so that's why you just did cardiac muscle. Excellent. So now we have the two proteins that can change the shape and generate the tension, generate the power. We have the two proteins that control it, turning it on and off. But remember, these proteins have to be precisely arranged. And to have these precisely arranged so that not only is it incredibly efficient, but it also gives the muscle its stripes, we need a third type of protein. And that third type of protein is a structural protein. And those structural proteins are what give the sarcomere its shape, its structure, which gives the myofibril its shape, its structure, which gives the muscle cell its shape, its structure, and so on and so forth. Now here, there are dozens of structural proteins found inside of a muscle cell. Some of them help this sarcomere align. Some of them hold them to other myofilaments next to this myofilament. And some of them, remember how we saw that individual muscle cells had precise arrangement as well? And that's because there's some special structural proteins that will actually go from one cell to the next cell. So there are dozens of these structural proteins, but I am only going to hold you responsible for 3. The 3 structural proteins you're going to be responsible for are the Z disc, titin, and Myoglobin. So these are the 3 structural proteins that you are responsible for. Let's start with the Z disc first. Let's see, I've used blue, I've been red, I've purple. Let's switch to, you can use brown. I like brown. Excellent. Oops, well, okay, that works too. Our Z disc is what forms the boundary of our sarcomere. It is the wall. Right here next to me, I have a wall. Not only is this wall helping to define the room that I am in, but it is also helping to define my daughter's room as well. And she's probably wondering why I'm knocking on her wall, right? But she's a teenager, so she probably won't care enough to come over and find out. But the point is, this wall is shared between her and me, and it forms the boundary of both. And that is what these Z discs do. They are these jagged shaped proteins that form the boundary of one sarcomere. So this sarcomere is from Z disc to Z disc. But guess what? Coming off of this at the end is yet another set of actin going off this way, and another thick filament of myosin going out this way. And a second sarcomere is forming that way as well. So the Z discs are the boundaries of the sarcomere. And not only are they the boundaries, but they're also the anchor point for the actin, right? Again, if I'm going to change the shape of the muscle cell, if I'm going to move, if I had a rope and I was pulling on that rope and that rope wasn't tied to anything, then nothing's going to change. So I have to have an anchor point for the actin. So notice as the myosin's pulling on the actin, it's going to pull on the Z disc, and that Z disc is going to get pulled towards the center of our sarcomere. And the same thing's going to happen on this side. And lo and behold, that sarcomere gets shorter. And when it gets shorter, my myofibril gets shorter. And when my myofibril gets shorter, my muscle cell gets shorter. And when my muscle cell gets shorter, it pulls on the connective tissue, which shortens the fascicle. And when it shortens the fascicle, it pulls on the perimysium and the epimesium, and the whole muscle changes shape. Notice these changes that are happening here at the molecular level is how I am able to contract that muscle and move it through space. These small little proteins interacting with themselves here is how we move our body through space. So having those Z discs forms the boundary of our sarcomere and allows us to change the shape. Now, remember, one of the things we talked about is that we need to be able to extend the muscle without damaging it. And then we also have to have that recoil to it as well. That comes from a protein known as titin. Titin comes from the Greek word titans, right? The Titans, remember, were the lords of the earth before the Greek gods took over. And there were these big, massive beings. At the time that titin was found, it was actually the largest protein that had been located in the body. And so that's why they called it titin, because of those titans. Titin is a big coiled protein. Let's use pink for this one. That serves two purposes. It has kind of like a slinky, a coiled end to it. This coiled end is what allows it to be extended without damage and also allows it to recoil afterwards. So this is what gives it the elasticity. This is what gives it its extensibility. And it also helps to stabilize the myosin and hold the myosin in place. So there is one titin on this side and one titin on the other side as well. And so that titin, oh, I spelled that wrong. T-I-T-I-N, titin, is what causes the elasticity, causes the extensibility, but also helps to stabilize and hold this individual myosin in place. However, and again, in simplicity here, I am showing one myosin filament and only 4 actin filaments. Remember, this is a 3-dimensional structure. Every myosin is surrounded by something like 8 actin filaments, and every single sarcomere is going to have, a myofibril is going to have multiple sarcomeres within it. So there's many, many of these. There's another one down here, for instance, below it, doing the same thing. So to precisely arrange all of these sarcomeres within a single myofibril, we have an additional protein, structural protein, and we'll use, running out of colors here. We'll just use black for this one. These long thread-like interwoven proteins, I'm going to just draw them as a straight line, but we'll see in a minute they are these interwoven proteins, and there are 3 rows of them. And these 3 rows of proteins, I forgot to write titin here, and this here, these are our Myoglobin. They arrange and align all of the myosin filaments, again, helping to give this precise organization to our sarcomere. I've drawn these here, but let's look at the pretty pictures from your textbook to make sure that this makes sense. As I mentioned, we have 3 types of muscle proteins. They are the contractile ones, the regulatory ones, and the structural ones. Here, we see our contractile proteins. Notice here, highlighter, we see a single dimer of myosin with those interwoven tails and the 2 heads. And notice hundreds of these are put together into that thick filament. right? That thick filament is just chock filled with proteins, which is where it gets its name as that thick filament. As I mentioned, the head of our myosin is a motor, where it uses the chemical energy from the ATP to generate a mechanical force, a mechanical energy, and that is a pivot of the head, where it basically grabs the actin and pulls. Of course, it has to have something to pull on, and so notice here in blue is that dimer, right? This individual actin subunits that have handholds on them. Those are the binding sites for the myosin head, so that the myosin head is able to grab and pull. But notice those binding sites right now are covered by our regulatory proteins. We have the long thread-like tropomyosin that blocks the binding sites, and the small handle troponin that can move it out of the way. Now, moving that tropomyosin out of the way sounds like kind of a bit of a wonky activity because it's going to allow the contraction to take place. So what do you think tells the troponin to move the tropomyosin out of the way? Haley's got it. There you go. Wonky equals calcium. Calcium binds to the troponin, changes the shape of the troponin, and moves the tropomyosin out of the way. I'd love some of these pictures from your textbook. This one here, again, using an electron microscope. Some of the technology of today is truly astounding. They're able to take a little bit of tissue from the body, freeze it, put it on a petri dish, stick it into these microscopes, bombard it with electrons. And when they do that, we're able to, from the refraction of those electrons, be able to deduce images of these tissues at a molecular level. And that's what we're seeing here. We are actually seeing one of these myofibrils at the electron level, at the molecular level. Notice we have these big, huge, thick filaments. So this is a myosin, and this is a myosin, and this is a myosin. And notice these myosin have their heads, that are coming off of them. And then in between them, we have those thinner actin filaments that the myosin heads are grabbing onto. And so we have this alternating pattern of myosin and actin and myosin and actin and myosin and actin and myosin and actin, overlapping, grabbing on and pulling to be able to produce that tension and contract the muscle through space. Here again, we see those regulatory proteins. The tropomyosin is the thread-like one, sits on top of the binding site, and troponin is the handle that controls it and moves it out of the way when calcium binds to it. And here we see a nice simple illustration of the structural proteins. Notice we have the Z disc, which is forming the boundary of our sarcomere and also the boundary of the next one as well. We have our titin, which is this long, again, thread-like. At the time, it was the longest protein that had been found in the body. This coiled, sprinkled, slinky-like part that can be stretched out without damage, with limits, and then will recoil back. And then it also aligns across the myosin. One point I want to emphasize is this from one to the left to the right is not one titin. Notice the titin molecules actually bind to the myomesin at the center. And that myomesin at the center is these interlocking. They've drawn it as a chain, but it's really these three interlocking proteins along the midline of our sarcomere, forming what we call the M line. We see these things here in this illustration. Notice this one doesn't have the regulatory proteins, but there's lots of other places where we can see this as well. Notice if we go back, here to our handout from the class, I told you we have a sarcomere model. Notice our sarcomere model, I love this, has a plastic cover on it. This plastic cover, though, isn't just a plastic cover. It's supposed to represent more anatomy. Notice here in yellow is the transverse tubule that wraps around the sarcomere. Here in green is my sarcoplasmic reticulum. And notice the sarcoplasmic reticulum thickens, gets bigger right next to the transverse tubules. So it represents the terminal cisternae. And so these collectively are the, let's make this a little bigger. Collectively are the triads. So we see the triad there. Notice here again, we see a couple triads, and notice from Z disc to Z disc, this is one sarcomere. Of course, we remove the cover. We can look at one side. Notice this big plate at the center represents the myomesin. These big thick filaments in the center are the myosin. These red are the actin. And here's our Z disc that the actin's anchored to. Notice also on the actin, we have these yellow thread-like proteins representing the tropomyosin and the little green handles, which are the troponin. We can see the heads of the myosin here and the felt in the blue. And notice the only thing we're missing is the titin. It doesn't have the titin molecule on it. But everything else we've identified, we've been able to see and visualize here. Excellent. So we see that here in that. So this would be what it would look like contracted, and then when it pulls out, it would be more of a relaxed state. As it moves and slides together, changes its shape. All right, so there are a couple pictures that show that nicely. The other way I want to show you this is to come back to this model. Excellent. Muscle model. Notice as we look at this muscle model, we can see some of this anatomy we have been talking about. No, it's T-I-T-I-N, 2 I's. But remember, you get one letter. So technically, if you spelled it T-I-T-A-N, you would get it wrong. So it's T-I-T-I-N, 2 N's, 2 I's. So that was my error. Okay, so here we can see in this model, in the myofibrils, we can see all of these proteins we were just talking about. Notice here, this is our Z disc. And in fact, from Z disc to Z disc would be one sarcomere. So notice we have one sarcomere represented here. Notice at the midline of the sarcomere, we have our 3 lines of proteins that are the myomesin. We have our thick myosin filaments. And we have our thin actin filaments that are connecting to our sarcomere. Notice this doesn't show the regulatory proteins, doesn't show the titin, but we see 2 of our structural, 2 of our contractile proteins in this and how they're organized. And again, we see that precise arrangement from myofibril to myofibril. This precise arrangement, and let's cheat and go back to my drawing of it. This precise arrangement of these allows, and like I said, forms the stripes of our muscle, right? We can see by the arrangement of these proteins how they're going to give it a precise shape and structure. So let's cheat and use yellow and use my highlighter. Notice this region here, from here to here, the portion that contains the myosin. Remember, the myosin is the thick filament. This region of the muscle of the myofibril that contains the myosin has dense, dark proteins in it. And this region is known as the A band. This is the part that is the dark stripe of our muscle cell. Notice this portion that goes from myosin of one sarcomere to the myosin of the other. This region here doesn't have any of that thick myosin in it. So this is what is referred to as the I band, and it is the light region. So these are the stripes. The stripes that are formed in our muscle cell are formed by the A bands and the I bands, by the presence of myosin or the lack of myosin. However, if you remember back, and I will show it to you again, when we looked at that slide that had the 4 muscle cells, remember when we looked close at it, we saw that there was a dark line in the center of the light stripe? Well, now we see what that dark line was. That dark line in the center of the light stripe was the Z disc. That Z disc down the center of the light stripe, because the light stripe covers portions of 2 sarcomeres. So it's got that Z disc, or what is called the Z line, right down the center. But remember, there was also a portion of the A band that was a little harder to see, but was also light. Because notice in a lot of, and here I am going to change colors to try to, what are we going to use? We'll use light blue. Notice there is a fair portion of the A band where myosin and actin overlap. But there is a small region there of the A band that just has the myosin in it, in the center, just has the myosin. This portion that just has the myosin, because it doesn't have myosin and actin overlapping, only has myosin, is the H band, or also sometimes called the H zone. And notice smack dab in the center of this, where we have just the, not just the, we have the myomesin and the M line together. These regions together are what is known just as the M line. I appreciate this vocabulary can get a little tricky. So one of the easier ways to see this, and notice our model does a great job of doing this, is it cuts the myofibrils at different levels. Notice, for instance, this myofibril is cut right here, just in the area where there's only actin. So notice in this area where there was only actin, that would be the I band. Whereas notice this one up here is where the myosin, oh no, actually, hold on. Undo. This one right here is where the myosin and actin are overlapping. So that would just be our A band. This one up here, notice, is just in the part where there is just myosin. That would be the H zone. And notice this one here is cut right on the M line, whereas there's myosin and myomesin. We can infer that by looking at it this way, but notice if we change the view and look straight down from the top, notice we can tell each of these regions by the proteins that are present. This one here just contains the thin filament, that actin. So what region, right? Notice we're looking at the regions of the organelle, which is exactly how I'll ask this on the exam. What region of the organelle is this one here that just has actin in it?
H band?
No, the one that just has actin, remember, is the I band. The one where the actin and the myosin overlap, that would be what?
A band?
A band. Remember, I will tell you right now that A band and I band were named after the German words because it was a German anatomist who first described this. However, if you notice, dark has A in it, light has I in it. So while that's not why they got their names, it is an easy trick to remember which is which. The dark band is the A band that's going to have the myosin in it. The light band is the I band that does not have myosin in it. Now notice right here, we have myosin, so this is part of the A band, but this part of the A band only has myosin in it. So this part of the A band that only has myosin in it would be called what? Yeah, this is where we have the H band or the H zone. And notice here, right on the midline, we have myosin and that interlocking myomesin. Again, there's myosin, so we're definitely in the A band, but this precise region in the A band would be what? Where there's myosin and myomesin?
M line.
This would be the M line. So notice with these cross sections of our sarcomeres, pardon me, sarcomeres of our myofibrils, we can actually see the proteins that are making up the different regions of this organelle. And your book does a nice job of doing this as well. If we go back to the lecture, right, we did all of that, we did all of that. And so again, notice, right, if we were to take our section through, right, the I band, notice we don't look at the titin there, we don't see the titin, but we would know it would be in there, but it would just be the actin. That would be the I band. Notice, oops, if we went through the A band, we would get myosin and actin. If we go through the H zone, just myosin. And if we go through the M line, we have that interlocking myosin and myomesin together there. So the different regions of this organelle is made up by different proteins. And these are so precisely arranged that it's what gives it its stripes. Now, I know these illustrations do a nice job of showing this, right? Showing all of these things does it really, really nicely, but it really does look this way. Here is our electron microscope again. Notice with our electron microscope, we are looking at one and a couple pieces of another myofibril. Notice here we actually see this stuff here, this bubbly looking stuff here is that Sarcoplasmic Reticulum and Transverse Tubule that is wrapping around the myofibrils, because remember those wrap around the myofibrils. Here we have that Z disc, here we have the Z disc. So from Z disc to Z disc, we have one sarcomer. Notice our myofibril has a light stripe containing actin with the Z line at the center. And then the dark stripe where it is comprised of myosin and actin. So this region that has myosin is our A band. However, notice it's a little lighter at the center, and it's a little lighter at the center because this small area right here is where there's only myosin. The myosin and actin are not overlapping anymore. But notice right at the dead center, we have that myomesin forming the M line. Here we see it with this nice electron microscopy view. Here's another one colorized. And again, here we see two myofibrils side by side. Now, one of the things I will point out to you is notice with this particular slide, we can see where the Sarcoplasmic Reticulum, we can see where the transverse tubules are. And notice they don't line up perfectly. Does that mean that they weren't perfectly aligned in the muscle cell? No, of course they were perfectly aligned in the muscle cell. But remember what they had to do to get this image. They had to take a chunk of muscle out of some organism. They had to freeze it. They had to slice it. They had to prep it with some chemicals. They had to put it in a petri dish. They had to bombard it with electrons. And so with all of that, you might have gotten some shifting of the myofibrils. But normally in an intact living muscle, these are perfectly aligned. And even with all that manipulation, you can still see it's still pretty close. Again, we can clearly see the region that contains the myosin, which is the A band, the dark band. We can see the part that doesn't contain myosin, that is the I band. We can see the Z line in the center of the I band. We can see the M line in the center of our A band. And we see that one little region that's a little lighter because it only contains myosin, and that is that H zone. We see it really nicely with the electron microscope. And if you remember when we looked at this at the beginning of class, we saw it even with a light microscope. Notice even with the light microscope, we were able to see the light stripe and the dark stripe, the light stripe and the dark stripe, the light stripe and the dark stripe. And notice at the center of the dark stripe, pardon me, at the center of the light stripe, we can see that Z disc at the center of it. And like I said, if you look really, really closely on some of these, you can see that it is a little bit lighter at the center of the A band. That is the H zone. Now, with an electron microscope, are we going to be able to see the M line in this one? No, so we don't see the M line at the dead center of the dark stripe, but we can see the H zone, the H band in that. And so it is this precise arrangement of these proteins that form the structure. And as we will see, they are also going to form the, or provide the function of this as well. All right. Questions on this? All right, now that we know the anatomy, we can start to understand how this is going to function. Our muscle contraction is going to be the longest physiological process we are going to study in this class. Notice I didn't say the most complicated, but I did say the longest, right? Because it is so long, we are going to take this muscle contraction process. And remember, we're still talking about skeletal muscle. So we start from the decision. I decide to contract that muscle all the way to the point where the muscle contracts. That entire process is the longest process we are going to learn in this class. To make it more easily digestible, we are going to divide it into three main processes. Now, obviously, we're doing this artificially because this is one long continuous process. But artificially doesn't mean randomly. We are going to do it in a meaningful fashion. We are going to divide it up into three main processes. And when we divide it into that three main processes, I guarantee 2 of these 3 processes will be essay questions on your exam. All right. So let's talk about the 3 ways we are going to divide it up. Step 1 is a process that is called communication at the neuromuscular, all one word, junction. Obviously, we don't yet know what a neuromuscular junction is, but we will have to talk about and identify it. And anytime we have a process, we need to know the goal of that process. The goal of step 1 is to convert a neural action potential into a muscle action potential. So step 1 is communication at the neuromuscular junction, and the goal of it is going to be convert a neural action potential into a muscle action potential. Step 2 is called the excitation, contraction, coupling. What does it mean to couple?
Join.
Yeah, to bring together, absolutely. Notice in step 1, we produced an electrical signal. We excited the muscle cell. But what we want the muscle cell to do is to contract. And so that's what we need to do. We need to bring those 2 things together. Yes, we have the excitation, but now I need it to contract. So excitation, contraction, coupling is the fancy name of step 2. but its goal is very simple, right? If we want the muscle to contract, what's stopping it from contracting all the time? Why aren't your muscles contracting all the time? 24/7 again?
You choose not to?
Well, okay, part of it is that we have that voluntary control, but remember, the myosin wants to grab on the actin all the time. So why isn't myosin grabbing on actin all the time?
Tropomyosin?
Yeah, those regulatory proteins, exactly. So our goal of step 2 is going to be to move the regulatory proteins. And what did I say we were going to use to move the regulatory proteins? Contracting a muscle is a wonky thing, so we need to use calcium to move the regulatory proteins. Excellent. So our goal of step 2 is going to be to use calcium to move the regulatory proteins out of the way so that step 3 can occur. Step 3 is the contractile cycle, right? One of the key words in that sentence is cycle. What does that mean? What is a cycle?
Cycle coming back, starting from an origin, coming back to that place.
Exactly. It is going to be a continuous process. It's going to be a continuous process of going around and around and around and around. Absolutely. We're going to get this cycle. The goal of this cycle is to generate our force, right? Generate our tension. That ultimately is going to be used to, so we're going to generate a force. Let's say it that way. A force. Generate a tension. And that is going to ultimately lead to the movement that we want. Now, this contractile cycle is also sometimes called the sliding filament theory, because it is the actin and the myosin sliding across the surfaces of each other that changes the shape of the sarcomere, and that generates the force and generates the tension. So I like that. Let's say change the length of the sarcomere, and that generates our force, generates our tension, and ultimately will lead to movement. Those are our 3 steps. Now, if there was a 4th step, what would that 4th step in this process be?
The relaxation?
Yeah, exactly. Once we contract a muscle, we don't want it contracting forever. If there was a 4th step, we could say relaxation would be the 4th step. But relaxation really could also be thought of as turning off the 3 steps. So if you think about it, for the previous question, steps 1, 2, and 3, we could really think of it in 2 ways. We could talk about how to initiate the process, and that would be the key way I would describe it on the exam. And that's, of course, basically how you start the process, how you turn it on. I could also ask you how to terminate the process, which would be how you would end that process. So we could think of relaxation as its own step, or we could think of initiating step 1, terminating step 1, initiating step 2, terminating step 2, initiating step 3, terminating step 3. Of course, if we really understand a process and how that process works, then we can actually manipulate it. We can modify that process, and when we modify that process, we see how it interrupts the normal function. So to truly understand these processes, do we also need to maybe know how to modify the process? Yeah. So if you think about it, there are 3 steps, but really, if you think about it, there are 10 possible essay questions. Initiate communication at the neuromuscular junction. Initiate excitation contraction coupling. Initiate contractile cycle. Terminate communication at the neuromuscular junction. Terminate excitation contraction coupling. Terminate contractile cycle. Modify communication at the neuromuscular junction. Modify excitation contraction coupling. Modify contractile cycle. Or if we wanted to, how do you relax a muscle? 10 possible essay questions. And as I mentioned, I guarantee you at least 2 of those essay questions will be on your exam. And here's the fun thing about the online format. Do I know which 2 you're going to get? Nope, because they're all there in a bag and you're going to randomly pull 2 of them out. Maybe 3. So this is what we're going to be doing. For the next couple weeks, we are going to be going through these 10 possible essay questions, these 10 processes, and make sure we understand it completely. Is it going to be hard? Is it going to be challenging? Absolutely. Are there a ton of resources to help you to be successful with this? Absolutely. If you have not been taking care of them yet or using them yet, you absolutely positively need to start. Like, for instance, your modified mastering A and P. In your modified mastering A and P, there are a tremendous number of great resources. Again, one of the easiest ways to find all the resources for a section is to go to that particular section. So like, for instance, muscle tissue or the muscular system, and do it that way. But I want to actually show you some specific tools that if you're not already using, first and foremost, shame on you. One of them is your practice anatomy lab. Hopefully you use this for your bones, but this can also be used for the muscles as well. So again, when we go to the muscular system, we can go to, for instance, the upper limb. We can look at the pretty pictures here from your cadaver model, looking at the labels, modeling it, looking through all the different levels, identifying the muscles. It can be a little tricky on here, but notice if you go back to the home page, while looking at the cadaver is fun, as you've learned, you're much more likely to be tested on the models. So instead, if we look at the muscular system and at the upper limb, do we see some pictures of some models with the muscles on them that can be identified and located this way? Absolutely. I can show the labels, I can turn them off. If I'm feeling particularly frisky, I can take a quiz where it shows me a muscle and I have to say, well, that of course is the deltoid muscle, and I can check my answer and got it incorrect, right? I can do that. And when I'm feeling particularly frisky, I can take a lab exam where it actually shows me a muscle and I have to write out what the answer of that muscle is. Now, how many muscles do you have in the body again?
Over 600.
Over 600, absolutely. Are you being held responsible for all 600? No, just the ones on the list. So if you happen to be taking a lab practical like this and it shows you a muscle you're not responsible for, what should you do? How about ignore it and move on to the next one, right? So that's something you can do. One other thing you can do to help you with this is under the PAL. Oops, where is it? Hold on, got too many windows open. Oh, I know where I want it. Here we go. Sorry, got too many open windows. I'm closing the chat, but I'll see if it. Notice up here, flashcards. Anatomical models, right? Notice I can go to muscles of the upper limb. muscles of the trunk, muscles of this, right? I can submit this. I can actually pick the muscles that I'm responsible for on the exam. And I can actually make my own flashcards where I can review the flashcards, seeing what the muscles are and identify them that way and go through all the list of these things this way and do it that way. Or I can actually quiz myself. I can actually make my own quiz from this as well. This is an amazing resource. As you're learning these, one of the things we need to do is we need to be able to recognize the movements. We'll notice also here on the top, we have animations.
Biceps brachii is the large two-headed flexor muscle.
Do you guys hear that? Okay.
Could you turn the captions on that for us, Professor, please?
Yes. Thank you.
Biceps brachii is the large two-headed flexor muscle located in the anterior compartment of the arm. The origin of the long head is from the supraglenoid tubercle of the scapula. The origin of the short head is from the tip of the coracoid process of the scapula. The two heads insert by a common tendon onto the radial tuberosity and by the bicipital aponeurosis, which blends with the deep fascia on the medial aspect of the forearm. Biceps brachii flexes the forearm at the elbow supinates the forearm, and weakly flexes the shoulder. It is innervated by the musculocutaneous nerve.
All things that you're going to be responsible for. On this exam, you need to know origins, you need to know insertions, you need to know actions of the muscles. And this does a great job of showing that. It isolates the individual muscles, shows the attachment points, shows the actions, mentions the innervations, which we're going to need to know when we get to the nervous system next. Great, great resource. So for the anatomy, this is something which if you haven't been taking advantage of yet, you absolutely positively need to start. All right. But there are a lot of other great resources on here as well. One of the ones that I love tremendously under the animations and videos is the A&P Flicks. Notice here we have our A&P 3D animation. This is another way to get to muscle groups, actions, origins, insertions, and actions, more animations of muscles and bones here. But what I want to focus on is this one here. Remember how I just mentioned to you, we've got those three big processes: events at the neuromuscular junction, excitation, contraction, coupling, and the cross-bridge cycle, right? So we have all of those, and these are these great high-resolution animations that do a great job of describing and showing these processes.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles.
Can you not see the closed captioning? Is it too low on the screen?
No, I can't see it at all. It's blocked by the pictures on the bottom.
Then there you go. Then I will do it. I'll keep it small so that we can see it.
An action potential propagates down a motor neuron to a skeletal muscle fiber.
Okay.
The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma.
And there you go. We have gone from a neural action potential to a muscle action potential, right? In 7 simple, easy steps with great illustrations, great animations that do an excellent job of showing these. Like I said, it's a long process, but each individual step isn't that challenging. But again, there are a lot of great resources. This visual one is excellent for those of you who are visual learners. There are art labeling activities, some things along those lines. But the other resource you may not have been taken advantage of yet, that is a really great resource that we can use for the muscles, we can use for the nervous, but especially when we get to 431 is going to be really, really important, is the interactive physiology. Notice there wasn't much for it for skeletal system, tissues, and chemistry. But notice for the muscular system, there is all these tremendous great resources. So again, with that neuromuscular junction, you can click on that contact and it will walk you through it. This is basically a directed tutorial that will walk you through the steps in an interactive process to help you to understand this material. While this material can be challenging, there is a lot of really, really great resources in here to help you to be successful to learn these. So if you're more of a tactile learner or a visual learner or an auditory learner or whatever your learning style is, we have the resources here that you paid for, I assume. So if you paid for it, make sure you're taking advantage of them. And of course, remember, this is also where you'll find your PhysioX that has those 7 activities for the muscles as well. All right, so that will allow us to play and manipulate with this and learn this material. So these are some of the resources that are going to help you to be successful. All right, questions on that? All right, so these are resources that'll help you with the physiology, absolutely. There's the physiology assignment that's on here, but we also have the anatomy stuff as well. So let's also not forget, there it is. As we go to the modules that we have here, this is also where your handouts are located, right? These are where the lecture slides are, where you're turning your assignments, where your handouts are located. Obviously, we have some muscle histology you're going to be responsible for. I think we've looked at most of that today, although we have a little bit more to cardiac muscle to talk about. We'll talk about when we get to the end of the muscle and a little bit more. But we also have our muscle anatomy list. So we have that list here. And actually, let me switch to this view. So here, that one, oops, I think I just closed that. That's not good. Oh, I did. That stinks. Hold on. There you go, that's what I want. So, just like we had for the bones, we had a precise list of the material that you were responsible for. We have the same thing here in our handout for the anatomy. Notice here are the muscles of the head and neck you're responsible for. 2, 4, 6, 8, 10. Do you think you only have 10 muscles in your head and neck region? No, you have a massive amount more of those. So this is all that you're going to be responsible for. Notice you are responsible for every muscle on the list and the actions of all of those muscles. Notice some of them just have one action. Some of them have two. Some of them have three. Some of the muscles on your list will have 4 actions on them. If a muscle has 4 actions, guess how many of those actions you need to know? These are the easy questions, folks.
All of them.
All of them. 4, absolutely. You need to know them all. Absolutely. You need to know all of them. All right. Notice also that some of the muscles on the list are bolded. Notice those muscles that are bolded, you are also responsible for the origin and the insertion. The attachment points for those muscles. Notice, for instance, a muscle like your platysma isn't bold. And notice it just has dotted lines here. Does that mean the platysma doesn't have an origin and an insertion?
No.
No, of course not. It means you're not responsible for it on the exam. So not only are you only responsible for 10 muscles of the head and the neck, you're only responsible for the origins and the insertions of 5 of them. So you're only responsible for those of this. One of the things that I find that is incredibly successful at learning this material are flashcards. I am a tactile learner, so flashcards help me because that when I'm writing them out, I'm often better at remembering them when I write them out that way. But the key to a flashcard for these is that too often when someone makes a flashcard, they just put the name of the muscle on one side, and on the other side, they have all the information, the origin, the insertion, and the action. That's not what you want to do. What you want to do with your flashcards for this exam is fold them in half and make them 4-sided. One of these days, I'm actually going to publish these 4-sided flashcards. I will be rich and famous. I won't have to teach anymore. But until then, you can take advantage of my vast knowledge, but to be successful in studying this by making 4-sided flashcards. So you fold your flashcard in half. You put the name on one side. You put the actions on the other side. Then you flip it inside out. You put the origin on one of those, and then you put the insertion on the fourth. So that when you're studying them, yes, the first time you study them, you can look at the name of the muscle or a picture of the muscle and guess what it is. But then you also have to think, all right, what is the origin of it? Or what is the insertion of it? Or what is the action of it? And the next time you go through, maybe you'll look at the origin and think about, all right, what muscle originates into the supraglenoid tubercle? And once you figure that out, then you can go, all right, well, what would its insertion be? What was its action be? It's a way of relating the material together because that's how you're going to be tested on it on the exam. On the exam, I can show you a model of an arm or a leg or something like that. I can point at that muscle and I can basically ask you those 4 questions. Identify the muscle, identify the origin, identify the insertion, identify the action. But you know what else I can do you? I can show you a picture of a bone and I can label a bone feature there. One of the bone features that you happen to have learned for the last exam. And I can say what muscle originates into this bone feature or what muscle inserts from this bone feature. And you need to be able to identify that. I guarantee usually somewhere around, I would say 10 to 20% of this exam is going to be bone features, pictures of bones, where you're going to need to be able to identify the muscles from their attachment points. Most of it will be models. right? Heads, arms, legs, those types of things, those resources that you have available for you. Again, when you're thinking of resources that are available for you, again, don't forget about not just the pictures that I have provided for you, but remember also we have many great online resources. One of them is that virtual anatomy lab. Notice for the virtual anatomy lab, we have a muscular system where we can see torso pictures. So it shows us that picture of a head, for instance. And then as we click through it, it shows us and identifies the muscles for them, right? Posterior view, muscles, right? Abdominal view, and so on and so forth. We have these great pictures that show this. Now remember, this is a global resource that was actually made by Cosumnes River College. So is every single muscle on here necessarily going to be on your list? No. So for instance, the pectinus, I'll tell you right now, is not on your list. Feel free to ignore that muscle. But is the sartorius, is the iliopsoas, right? Is the iliacus, is all of these on here? Absolutely. So these are ones that are on here you're responsible for. So it's a good resource. Does a great job of showing these things for us. We can see lots of great resources, as well as, I didn't like the leg models that they had on this as well. So I used, I added some additional leg models to your handouts on the modules as well. So make sure you're learning these, be able to identify them on models and charts. Bodybuilders. There will be 2 to 3 questions where I will show you pictures of bodybuilders, and you have to recognize superficial muscles on the bodybuilders. And of course, there's going to be histology as well. So I think that's pretty much everything that will be on this lab exam. All right. So there are things I'm going to do to help you to be successful. One of the things that we will be doing, as I mentioned, is the muscles that you're responsible for the origins and the insertions for. We will actually go back and draw them. Because one of the advantages that I see is that when we look at a muscle and where it attaches and which joint it crosses, it helps us to be able to know what the function of that muscle is. So there is a handout on Canvas for you where we will be working on this and drawing. For the next class, you need to have a red and blue crayon or marker or pen or pencil or something. Because by convention, and remember, by convention is just a fancy way of saying a rule that a bunch of anatomists got together and made up. Red is the color we use for origins, and blue is the color they use for insertions. So we are actually on this handout, print out this handout, have a red and a blue pen or pencil or marker, and starting on Monday, we're going to start going through the muscles, drawing the origins, drawing the insertions, identifying the muscles, and identifying their actions. All right. And there's one more thing. As I mentioned, there. Like I said, a muscle like the masseter that just elevates the mandible isn't too challenging to recall. However, as we go down the list, like for instance here on the leg, how many actions are there for the sartorius?
Four.
There are 4 actions for the sartorius. What are they?
Abduction, flexion, and lateral rotation, thigh flexion, flexion of knee.
And if you write that on the exam, yeah. So again, you got to be careful. When we talk about, actually, let's start up here first. When we talk about the action of a muscle, there are 2 ways we can talk about it. We learned the vocabulary in the last section. Flex, extend, abduct, adduct, and so on. When you talk about an action like the bicep brachia, this action right here is a flex. And when you're describing that, not only do you need to say that that is a flex, but you need to say what it flexes. And there's actually two ways you can do that. You can either identify the joint that it flexes, so it flexes the elbow, or you could say the part of the body it moves, flexes the forearm. So flex the forearm, flex the elbow, they mean the exact same thing. But notice you can't just say flex, you have to say what it flexes. And it's the same thing here. So when we talk about the actions of the sartorius, one of the actions is to flex the hip. One of the actions is to flex the knee. One of the actions is to abduct the leg or hip. Again, I could say it either way. And the other is to laterally rotate the leg. Now, one way you can memorize that is just with brute force. You can just brute force memorize what those four things are. However, what could be more convenient is if you can put them together into a common activity. So notice if I were to flex my hip, flex my knee, abduct my leg, and laterally rotate it, by doing those four activities, I have crossed my legs. So that ability to cross my legs is because I flexed my hip, flexed the knee, abducted the leg, and laterally rotated it. So now I have a common activity that is going to help me to remember the four actions of the sartorius muscle. And that's your job. You have a handout that is going to be graded for correctness. Oops. Oh, I don't have it pulled up. So let's go back here. Man, where'd all you go? Oh, I'm not sharing, that's the problem. Got it. So I want to be here. And now that I'm here, notice there is a muscles activity homework assignment. For that muscle activity homework assignment, you are going to be doing two different activities. One thing that you're going to be doing is I've given you a common activity, and that common activity is described, and you're going to tell me what muscle is responsible for it and what the actual actions are. For the second half of it, I've given you a muscle and its actions, and you are going to give me a common activity. So notice we just cheated. I gave you, I thought I had the sartorius on here. Nope, apparently not. All right, but if the sartorius was here, those four actions would be listed, and then you would describe crossing your leg as the activity that is going to help you to remember this. This is a graded assignment you are turning in for a grade, graded for correctness, but you're also going to be demonstrating some of these activities. So I want you to be thinking about the muscles as we go through this, because I'm going to ask people to demonstrate some of these activities as we go through it. Yes.
I have a question regarding the second portion. As you said, the activity portion, can you go down? Like the muscles is there, the actions are there. So what activity? Like, for example,
again,
for instance, as we were going for the buccinator.
Well, but let's not use one that's actually on here since it's a graded activity. Let's do this instead. One of the things that could have been on here and apparently isn't is the sartorius. Right, the sartorius would be here as listed as muscle, and as I mentioned, well, actually, let's do it this way. It has four actions, right? Flex the hip, abduct the hip, flex the knee, and laterally rotate the hip. Those would be the four actions that would be listed here for the sartorius. And as I told you, the activity that you would do that would incorporate all of those things would be crossing your legs. All right, so that would be the activity that is going to help me remember those four actions. right? If we
President.
go back to the masseter, I mentioned the masseter elevates your mandible. When do you elevate your mandible? What's an activity that you do when you elevate your mandible?
Open my mouth.
Chewing.
Well, remember, elevate would be closing, right? Chew, or really think about it, that first bite. As you take that first bite, right, that's when you're elevating your mandible. Chewing, I always think kind of like, you know, your jaw's all over the place as you're chewing that cud. But that very first bite, you're elevating your mandible for that. So again, if we needed a mnemonic for that, with one action, we might not need a mnemonic, but if we needed something to help us to remember what the masseter did, taking a bite would be that common activity that would help us to remember it.
I think it's not easy, sir. It's really challenging. Like,
yeah, well, anatomy and physiology is hard.
To say the activity, to explain the activity, like having all the actions and say the activity, it's not easy, sir.
I appreciate that, but there are lots of different possible answers. So your job is to see what a muscle does and to come up with some activity that helps you to be able to remember what it is. Allison, we'll get to that one when we get to the muscles on Monday. I'll tell the story behind that one.
Sounds like a good story.
It is. All right. Any other questions? So these are the things, and like I said, we'll be working on this together as well. So again, I encourage you to get started on this ahead of time. You have 5 days before we meet, so you can start working on these on your own. But we are going to work together as a group to come up with some ideas for these things that are going to help you to be successful. Again, because that's the goal. The goal is to help you to learn this material, and I'm trying to provide as many ways as possible to be able to do that. All right. Questions on that? All right, well, that should keep you busy this weekend. So I will leave you guys to that. Do not forget also that you have that Labster that is due on Monday as well. Start taking a look at the resources and the materials that are on here to help you to start to be successful. And that is all I have for you. Have a good weekend. Be safe, be smart, listen to the doctors, not to the politicians. Enjoy the debate tonight. Hopefully it won't be quite the train wreck that the first one was. Like I said, be happy, be safe. Register to vote if you've not done so already. If you're voting by mail, remember today is when they recommend getting it in the mail to make sure it gets there on time.
And Sarah, what would be expected the grades to be posted for the exam, previous exam? When?
So usually it takes me about a week to grade things. I'm about a third of the way through the lab exams right now. Again, with teaching, I've got a class tomorrow as well. So usually Friday and Saturday is when I'll get the bulk of my grading done. So my guess is that probably sometime by Sunday, I'll get the grades posted. And then, like I said, on Monday, like usual, you guys will get the chance to view it. So again, it takes me usually about a week. So that's what I anticipate in this case.
I think in the beginning of class, you had mentioned that there was some kind of extra credit course that we could take, like a study course.
So yes, there is. So if you're not able to do the open lab on Fridays, there is a Science Success Center that you can find. There's a link on the first lecture. It talked about it, but I don't know what their timetable is. It's a 6 to 8 week half unit class that can be taken to help you to be successful. I don't know if they're still taking enrollment, so that could be something that you look for here. Let me do this real quick. So hold on. That's not it. Here we go. So if you go to the website and do a search for the Science Success Center, Here is the information. It's basically a time management, reading textbook, notebook taking class. So this is an activity that can be done. The contact information is here. You can contact them. I'm not sure if they're still taking students for it at this time, but if you complete the activity, you can get 15 points of extra credit for doing that as well.
And the second extra point was, sir, for 20 points. Was there anything for 15 or 20 points? Another one?
No, both the Science Success Center and the Open Lab can be, you can get 15 points of extra credit, but you can't double both of them up. I would love to be able to give extra point for voting, but I can't do that. I can strongly encourage you. You'll have a warm place and a cockle in my heart if you vote, but I can't actually offer extra credit for that, just because for the people who aren't able to. So, but I would love to be able to do it, and I want to strongly encourage people to be able to do it. For those that have already voted, exactly, excellent, congratulations. But no, I'm not able to do that. But voting is your duty. You guys are the future, right? You should definitely be able to control the world that you live in, and this is your opportunity. Don't let old people like me who are close to death decide things. You guys, you are the young, you are the future, right? I believe that children are the future. Lead them well and let them take the way. Show them all the beauty that you have inside. All right. I made you laugh. I saw that. All right. Excellent. Any other questions? All right. Excellent. That is all I have for you today. Like I said, have a good weekend, and I'll see you guys on Monday.
Sir.